Director, ICAR-NIVEDI, Bengaluru. Dr. Aniket Senior, sir, Director, ICAR-NIVEDI, Bhopal. Dr. Pallav Chaudhary, sir, Joint Director, IVRA, Bengaluru. Dr. B. Srinivas, sir, Head, Southern Regional Station, NDRA, Bengaluru. Dr. S. Hilary, sir, Executive Director, BioVet. Dr. Y. Malik, Dean, College of Animal Biotechnology, Veduvasu. Dr. Naveen Kumar, sir, Principal Scientist, ICAR-NRC, Quay. And Dr. Y.K.M. Reddy, sir, Retired Professor, Tanua. Dr. B.R. Gulati, sir, Director, ICAR-NIVEDI. Dr. D. Hemadi, sir, and Dr. G.B. Manjunath Reddy, sir, the organizing secretaries of this training program. And all the dignitaries who joined by online mode. And the most distinguished participants from different parts of the country, respected scientists of this institute, research associates, and dear friends. A very good morning to all of you. We are really fortunate enough to have you all here.
हमारा नहीं
There was some problem with audio.
आ रही है ना आवाज? हाँ जी, ओके ओके,
अब आ रही हाँ
ठीक, keep on continue.
So let us start today's training program with ICAR song, and may I request Ms. Arpita to play the ICAR song.
जय जय कृषि परिषद भारत की सुखद प्रतीक भारत भारत की कृषि धन पशुधन वैज्ञानिक विधि शस्त्र श्यामला छवि भारत जय जय कृषि परिषद भारत हिंद प्रदेश शिक्षा आशा स्वावलंबित भारत जय जय कृषि परिषद भारत जय जय कृषि परिषद.
Thank you, Archita. Today we are having with us Dr. R.K. Singh, sir, President, Indian Virological Society. May I request Dr. R.K. Singh, sir, to address the gathering, and sir will be talking about IVS perspective on Lumpy skin disease.
Thank you. Good morning, colleagues. Greetings from Indian Virological Society. Honorable Dean, Medical Science, Dr. B.N. Tripathi, Dr. Abhijit Mitra, Honorable ICAR-NIVEDI Commissioner, Dr. B.R. Gulati, Dr. Nivedi, Dr. Bhatta, Director NINP, Dr. Sanya, Director NIASAD, Dr. B. Srinivas, Dr. Pallav Chaudhary, Joint Director IBRI Bengaluru, Dr. Som, Dr. Pilari, Dr. Y. Malik, other dignitaries who have joined online, all the faculty, participants offline and online, colleagues. So at the outset, I take this opportunity to thank Dr. Gulati for giving me this opportunity to be here amongst you. Please, as we know, when we got Lumpy disease in 2019, it was a very difficult situation. We were in a situation where we were able to go for diagnosis easily, but then there was, how to control the disease. And at that point of time, based on the literature, we went for finding out a solution which can be immediately used as a relief measure in the country. And accordingly, based on the literature, published literature, Both-pass vaccine was thought of as an alternative. We get a new vaccine. At that point of time, groups also started developing vaccine, and currently now we have a vaccine which is, I think, at quite advanced stage. But now the situation has been, I think, it is much better. I didn't see the latest data, but there had been improvement in the situation. But then we are still not out of the challenge. Challenge will always remain. So in the current situation, 2-3 things they come to my mind as a scientist. Like there are situations now, and I think we already have a project on epidemiology of COVID in NASL. So I think that would be the good project wherein gene activity they can be taken out. So I could post these points. Now there are 3-4 scenarios. Number one, there are animals where both-pass vaccine has been used. There are animals where Lumpy new vaccine has been used. Then there is also a situation where 3-pass vaccine has been used. And some people they also got measles vaccine that is also used. Another scenario is where no vaccine has been used. So because there is a tendency of development of recombinants in this virus, so it would be a good research activity when we take up the genome sequencing in these areas and try to see that which is the situation where there is either no generation of recombinants or there is a possibility of having some recombinants. So that is one perspective. Second perspective is this vaccine is already live attenuated vaccine is developed. Data is being generated and it will be there. But then same time, we need to develop deletion mutant vaccines for longer run. And here I think rather than going in by gene, it would be good because in lumpy skin disease, there are 9 genes which are activated, which are being expressed and they are responsible for giving the changes in clinical symptoms. These genes, they are silent in both-pass and 3-pass, the other 2 viruses, which are in the Capri-pass virus group. So I think developing deletion mutant vaccines by deleting the genes would be a good approach. So these are some research challenges, but of utmost importance is the first point what I mentioned to generate, because we are in a situation where we have a live situation. Earlier we did not have disease. So once we already have a challenge, why don't we take advantage of the challenges what we are already facing, trying to generate plenty of data. Now these vaccines have been given. So another advantage we should take up, animals which are already vaccinated at the organized farm, data vaccination is already available. So we should collect serum from them and try to assess what is the duration of immunity. So when we are in such a situation, it's better to generate as much data as we can, either in terms of epidemiology, genomic, of course, by genomic sequencing. And then, of course, on the duration of immunity of the vaccine. Because now, initially it was 2019 when vaccination started. So now 2 years, 20, 21, 22 already gone, 3 years. So now they are good subjects to draw the serum samples and then see what is the duration of immunity. That will also give a lot of, because now there is a lot of demand of vaccine. When it is being produced also, there will be a lot of demand. But now if we know that there is already persistence of immunity and it is 2 years or 3 years of immunity, then at least those animals can be avoided, fresh animals can be vaccinated. So we need to develop data on all the aspects. And I think this is one of the greatest challenges. So throughout the day, we'll be here debating. I think now DD is very busy today. So I make this brief presentation and then we'll be interacting during the whole of the day. Thank you for your patience here. Thank you so much.
Thank you so much, sir. Now may I request our Honorable DDG Sir, Dr. B.H. Tripathi Sir, to address the gathering. Sir, please.
Thank you very much. I am audible.
Yes, sir. Yes, sir.
So my distinct colleagues, very senior colleague, and of course in the ministry, Dr. Mitra is here. Mitra has joined.
He is going to join in 5 minutes, sir.
Okay, so Dr. Mitra he will be joining, Environmental Commissioner and my esteemed colleague, and my senior colleague Dr. R.K. Singh, President IVS and former Director IVRI, Director NIVEDI, Dr. Gulati, and I can see Dr. Bhattacharya, Director NRC, and Director Nisad, Dr. Nikesh Sanyal, and the Director NINP and Bangalore, Joint Director IVRI Campus Bangalore, Dr. Pallav Chaudhary, Dr. Tilari from Biovet, Senior Executive Director, Dr. YPS Malik, and many senior officers from different universities and institutions, and very distinguished speakers who have been working on LSD, and all your participants from various institutions and universities. So this workshop I wanted to organize, and I thought that I will be in person there, and the program was like that only. But due to an important meeting here in ICR chaired by DG, I had to rush back and prepone my visit. I thought I would be there for a full day till the entire workshop is over, because I had a certain objective to organize and participate in this meeting. So the topic is very important, and I suppose that all participants and also distinguished speakers, they are taking part in this conference, this workshop. They are really, have worked a lot and give their opinion so that we can take further action. I suppose Dr. Mitra will join soon, because in fact, I planned this workshop only for the government to have a necessary action program, that how we should go along. And here a few messages I would like to give, that now, due to globalization, even the disease in far off places like African countries can come to India also. We have never thought, at least I have never thought, that during my lifetime, in my service period, these two important diseases will come in the country. We've never thought this lumpy skin disease, second ugly disease, which we are seeing photographs during our graduation and post-graduation at IVRI. Just reading only for the sake of differential diagnosis, and both diseases, the African swine fever and lumpy skin disease. But both have come and they have spread throughout the country. So any disease found in any corner of the country, any part of the world, is not far off from you. That can come at any time, and therefore our preparedness, current knowledge and technology must be leveraged as quickly as possible. We are already struggling with not many endemic diseases. Government has not got rid of still FMD and brucellosis and PPR and classical swine fever. Another very two important disease, one is a chronic one and another one is the acute one. Two different type of disease, two different types of animals. Now we have accepted in the country. And the country can't afford now. With such a competent manpower, such a huge veterinary infrastructure in the country, India cannot afford to accept any more disease. Still, we are spending almost 14,000 crore for control and eradication of 4 diseases that I named just now: FMD, PPRCS, and brucellosis. Now, another 2 diseases are on the head of government to tackle with, and also state government. So our thinking, our approach, our conventional and traditional thinking will not serve the purpose. Whatever we have today, and I suppose that India has a competent manpower, competent scientists, competent veterinarians throughout the country. If we are really on it, we could have controlled it. These 2 diseases. I will not say that lumpy skin disease, because it's a chronic one, the setup is insidious. We do not know then from where it has come and how it is spreading in the population. A bit difficult one, but since it has been circulating in India since 2019, we did not take care. We try to hide it, but we cannot. Even as a responsible person, we try to hide it, but along with that, we have taken some attempts, some measures to control it then and there only, and we have not realized it, that how dangerous this disease is. And similarly, in the case of a quite entirely different disease, acute event, I'll call it ASF. We had a chance to finish it in North System region itself, but we could not. I'll say. Therefore, one thing that has come out during the last 2 to 3 years, that we cannot be complacent. As a scientist, as a technocrat, as an expert person, we always give correct advice to the government so that government can really take the right kind of step, and that is required. So friends, if you say that but very recently, I'll tell you an example. Very recently, I was in Maharashtra. And for the release of, they were taking the vaccine, Maharashtra government, and they take at utmost priority, where CM, Deputy CM, and Honorable Minister Sirupalaji was also there. So many people that too, they did it in Vidhan Sabha. I'm audible?
Yes.
So that is taken such a priority. And then they submitted me data also. They submitted me data also that and see, he's the first state that has vaccinated entire animal live with the cattle population of around 1.4 crore. 140 million population they are vaccinated with goatpox vaccine. When I asked the other data, he said that around 418,000 were the infected cases, and out of that, roughly more than 30,000 died. So the question is that we did a good thing. ICAR advised that we had to go for the goatpox vaccination immediately. That was based on the literature, and that was an excellent decision that had to be done, and it has resulted in creating some immunity in the population, and therefore some disease incidence has also come down. But when we also look at the data of organized farms, even our own institutions and university farm, we found that disease has come after 6 months, 7 months, 8 months of vaccination with goatpox. So certainly it is not, it is going to be a foolproof method that you can protect. And it cannot be alternative for the vaccine. Fortunately, we have got the vaccine. Fortunately, I would say. And I would really, see that some initiative was taken by NRCE, not as a part of vaccine development initially. I was there itself, so I'm telling the story of 2 minutes at a time I'll take. It is just for the purpose of having a virus in our repository. And normally, virus is not shared by any virologist of an institute unless they thrash out. They all work. And when they feel after a few years, they might submit to NCVTC. That kind of attitude and approach should also be changed in the minds of our scientists. So it was just a matter of chance that I got some invitation to send my team, and we got the virus from there. And after that, we when people were in the COVID period, it's all COVID period, this has been done. People were doing their, looking at their health, other thing. Scientists at NRC was doing a good job in the lab. Somehow we got the very good candidate that has worked. ICAR came forward for this testing, the challenge, etc. at ICAR, and we got a good result. And I think more than 1.5 lakh doses of vaccine has already been circulated. I mean, given to different state government, like JNK, MNP, and many other Kusala and others. Results have been pretty good and satisfactory. And testing is going on, and companies have taken that. So that's one thing. And another question that came quite frequently for this virus was the recombination. We also read it because the vaccine available, 2 vaccines available throughout the world or other parts of the country, they had some reaction. They always saw some needling reaction, and they were not very good. And then that has perhaps led to, and also some papers, the spurious batches were there, which contained even all 3 viruses, sheeppox, goatpox, and LSD has always been a subject of study in African countries only. The European and American, they have never studied this because the disease was not there. And therefore, there has been a lot of controversy and also a lot of, and questions on the vaccine were there. So certainly that has led to raise questions against this vaccine as well. But anyway, that things are over now, people accept it. So the vaccine is working well, absolutely, there is no doubt, I think, and testing is going on, the result will come, it's safe also. Why recombination is being raised for this virus only? LSD virus. Here virologists are sitting there. They will trying to get some answer out of this. Even RNA vaccine, we have attenuated vaccine. Recombination is more possible. More the possibility of having recombination of live attenuated vaccine in the field population with RNA virus than the DNA virus, which is a quite a secured virus, quite big virus and quite stable virus. So virologists have to find answer why there was so many questions for this Capri virus than even for goatpox and sheeppox. Or let's take other live attenuated vaccine. There are so many live attenuated vaccines are there in the field. And has there been any evidence of either this DNA virus or any live vaccine that has gone back to virulence? So this workshop should churn out and get some answer of that. That recombination is simply a fears only, or it is real for this virus. If it is real virus, we have to work on that and look at this. Capri viruses had not been really worked much, and this is high time that India should take the lead and work on this virus. Not only point out vaccine is over, vaccine is made. Other aspect also. There's also some genotypic changes, some gene mutations also, as reported by some other scientists from CSIR. But we saw that is also not a new distinct serotype that is not being protected by the current vaccine. So this works out. I would also like that there should be a consortium that can also go further, looking at the genotype and gene sequencing of viruses from different parts of the country. So that part we should also take care of as a scientist. If at all some new viruses or new serotype or new type is evolving in the nature, in the environment, in the animals, that we should be able to detect that. And that is our duty of ICAR and the scientists, that we should keep a strict vigil over that. And for that, I'll try to make a consortium of the scientists, including some of the biotechnologists from Gujarat or so that we can have an answer to that as well. Now, other than the epidemiology, hello? कब से ड्रॉप हो गया? तो अब आवाज नहीं आ रही है? अब आ रही है आवाज? अब देखिए, अब आ रही है, पता नहीं क्या बात है, क्यों दिक्कत आ रही है? अच्छा, तो मैं दोबारा लगे करूं, फिर से लगे करूं मैं दोबारा? अच्छा, ठीक। Now am I audible?
Yes, sir.
Audible है
No.
सर, you are audible सर.
नहीं, डॉक्टर गुलाटी ने कहा कि कुछ प्रॉब्लम हो रही है वहां पर, आवाज नहीं जा रही, तो फिर दोबारा मुझे लॉगिन कर रहे थे वो।
हम लोगों को सुनाई दे रहा है सर.
हमें तो प्रॉब्लम होगी।
अच्छा, उनके यहाँ प्रॉब्लम हो रही है?
उनका प्रॉब्लम है सर.
कोई प्रॉब्लम होगी क्योंकि यहाँ सबको सुनाई दे रहा है।
अच्छा, वहाँ बोले था उनके कवरेज प्रॉब्लम है ये। उन्होंने कहा मुझे इधर आप रुक जाइए आप, तो हमने कहा मैं रुक गया इसलिए। अब हमें लगता है वही मतलब आउट हो गए हैं द्विवेदी वाले।
पूछ लेते हैं सर, उनको फोन करके पूछ लेते हैं।
हाँ,
वो सर अभी वो उनके नेटवर्क का कुछ इशू हुआ था, वो दे हैव रिजॉल्व्ड विद इन अ मिनट और टू, दे आर गोइंग टू लिंक अस।
हाँ ठीक ठीक ठीक
स्टार्ट कर दीजिए सर।
ठीक ठीक है. So thank you. I don't know. I was perhaps discussing about the genomic consortium to keep a strict vigilance over the virus, because it has been reported that based on the 6-7 samples, they have been able to find some mutation, but that is not a significant mutation to change the virus. But even then, as a precaution, we must now see that from different parts of the country, virus sample must be tested, and also must be sequenced to see that virus is not changing. And then again, the question comes that why it is changing with this Capri virus only. Again, the biologist must find answer to that. Excuse me. Then another thing that I would like to epidemiology of this virus. Still, I request maybe or other people to go for the epidemiology. Now, there is always a craze for the vaccine development or diagnostic development. But there are many things that is still left of the other viruses, that we are not able to study because craze is not there to study the epidemiology. You may not give much of importance, but really those data, basic data, is very important for the virus, and it is the least studied Capripox virus because it has been a common disease in African countries, where we never expected any such like advanced study to be done on this aspect. So my request with all my 4 or 5 animal health institute that this would work in this direction and try to work in a network mode. Even if ICR doesn't work, that doesn't have a network program, but you yourself can, because this workshop has been called for this purpose only. How we can work together also for different aspects, and nothing is left out of this. So then coming to the some diagnostic tests. Now, since disease has already gone to many states, maybe more than 20 states, and for the time being, it has come down because of the winter season. It will come again as expected because of some vectors are less during this period, and therefore it has come down. So, and this is a chronic disease where wherever it has gone, they have been successful only by the homologous vaccine. So that part we have to keep in mind, that if goat pox or sheep pox vaccine is not providing complete protection, then perhaps we need to really think of in that direction and adopt our homologous vaccine as quickly as possible. Then coming to, before I congratulate Dr. Naveen and his group for this virus, for this vaccine, and working intensively throughout the COVID period. That has been a great attempt and effort and commitment of NRC and VTCC to come out this vaccine. Another aspect, we need to have a diagnostic test. And of course, I have come to know, and I've discussed with that 3-4 institutions already have developed some diagnostic tests. NISAD also, NIVEDI also, ICAR-NIVEDI also. So we need to adopt one of them for our next selection program. And that is very important. Because when we start a next selection program, a next control program, where unfortunately it could not become a central sector scheme, though it was submitted by the government, by Department of Husbandry as a central sector scheme, fully funded by center for vaccination of animals throughout the country. But perhaps I came to know that they have given 60-40. So even then the state will come up and government will also see and make a surveillance. So when we start any program on the basis, as we have been doing it for the 4 diseases like FMD, PPR, and others, we need to have a next selection program. And this meeting specifically I called for this purpose, that all our 5 institutes are here, Animal Health Institute. How we have made a program for PPR and CSW, etc., and of course FMD as well. Similarly, on the same line, I want that the programs will be devised that who will do what, zero surveillance, zero monitoring. So we need a test for zero surveillance. Now we want test for zero monitoring as well. And that we do not have because capillary viruses are so similar, it is difficult to find a specific test for lumpy skin disease, a virus, and also have a kind of DIVA test. Now, once the program will go on, then we have to really detect that whether it is infection or the vaccination, antibody. So for that, it is also a challenge for the virologists that they should sit together. All people who are working on this, perhaps they are in NIVEDI itself, so they should sit together and they should take the virus, the vaccine virus, and trace out that can we have some test, some physiological test to differentiate between infected and vaccinated one. So that part has to come out. And a complete excellent program that who will do what in all these 3 or 4 institutions. They are participating in national action program for control education of LSD. That should be chalked out there, and we can have another closed meeting, a physical meeting, where we will finalize it. But whatever scientific issue, technical issue that is related with the vaccine, with the diagnostic test, and maybe recombination and other issues, if they're coming out, there must be consensus that, and accordingly we'll proceed ahead. So I think I have already taken more than 20 minutes. I'm not going to speak more, and all the scientists and all of the distinguished speakers, Dr. Naveen and Dr. Klari is there, Dr. Manjunath and Amit, and also from Nisad Mishra. So all people are there. So they will sit and they will discuss the issue. They should discuss the issue in good faith of science and technology, and so that we can come out with a concrete action plan. Now, another message that I would like to give you, all of you, that now traditional and conventional thinking has to go. People will not wait. People will not wait. We have not substantially progressed towards the ASF vaccine, though NISAID had adopted different approaches. The day virus entered, it would be the best if we start working before it comes to country. It introduced in the country. That is the first approach. If that has not been possible in our country, we are not able to import viruses and look at and work on that, preparing. Only for preparing diagnostic tests is not a solution. And then what we see normally, when such like these, we know that ASF, for last 2-3 years, we have been just discussing this program is being made, that we have developed this program is made. That will not work, and that has not worked. There has been for 6-7 months the disease was confined to only North Eastern region, and it has gone to entire country. Where is the problem? Where was the problem? I said earlier, therefore we can't afford. And the entire pig industry is devastated already. We have not learned lesson from China and many other countries. We had an opportunity and chance to finish it in last year also, when it was only in few districts of Assam and Arunachal Pradesh. Now again, see the same traditional process. Vaccine will be prepared. The government will have a plan for that. They will fund it. So those things will not come. Now, not acceptable. Everything is counted on in terms of economics. Now we are losing, and we know that pig is beared by who are the people who are rearing pig. How it can be socioeconomically important disease. Even our farm NRC pig has been finished. I don't know whether the direct NRC pig is here or not. So entire population was vanished. So you can just assess the amount of losses the country has been incurring due to ASF. We can say that it is a very complicated virus and vaccine has not been successful in any part of the world. But you are the competent people. How can we can develop it? We could have started our attempt on day one. The moment we had a virus in our hand, we started working on that itself. We should not wait for the project will come, this and that. And now people due to COVID, vaccine, there are so many platforms within 6 to 1 year. Number of vaccine has come up in the world, different parts of the world. So people know it now. The technology is there. Our stakeholders, they know it, the scientists can do it. And therefore the expectations are very, very high. And we have to come to the expectation of our stakeholders, our farmers, our people, others. Company is always there. Some Dr. Clary is here, Biovet is here. They're always ready to take the technology. And even you have seen that ICMR, when another disease came to India, they just give the virus only by who will come forward to make the vaccine. Because the people is not going to wait for you. That you will prepare vaccine after 8 years, 9 years, then the government of India will run a program and there's nothing doing. You have to change our mindset. We have to change the expertise of our scientists and our technology. There's also be leverage to come up with the product immediately, technology, so that can solve the problem. So with this, I'm not going to speak some of the issues that I have raised. Those issues should be discussed, and we should come out with a concrete answer and plan of action for that, so that we can accordingly work on that. With this, thank you very much. And I'm sorry, Dr. Gulati, that I could not be physically present there due to today we have the EFC meeting, important meeting. I have to prepare for that. So with this, I thank all the people who are working on LSD and Dr. Naveen, Consolation, Amit, and Dr. Mishra, and Dr. Manjunath and his group. All these 4 or 5 people, they are working there. And they will come up with the expectation, and will try to finish the disease as early as possible in 5 years, 5 years time. That is the target for you. And also for the government, if Dr. Mitra has joined. Thank you very much.
Sparing your one. We are pointing out about the different aspects that need to be discussed today for the control of LSD in our country. Thank you so much, sir. Now we are having Dr. Abhijit Mitra, sir, Animal Husbandry Commissioner, DSP, Government of India. May I request Dr. Abhijit Mitra, sir, to address the gathering. Sir, please.
Yeah, very warm good morning, our Honorable DDG Animal Science, Dr. Tripathi, Dr. Gulati, Director of NIVEDI, Dr. Bhattacharya, Director of NRC Equine, ADG Animal Health, Dr. Ashok Kumar, Jyoti Mishri, and I could see many renowned scientists and stalwart who have worked in the area of biology as well as the particularly of LSD. I could see Vice Chancellor of Kavashu. I might have missed some name. Excuse me for that, because I don't have the list of all the participants with me at present. Very warm good morning, and it's a very timely initiative taken by NIVEDI and Dr. Gulati and his team to discuss and deliberate on the issues on LSD. So far, our Government of India initiative is concerned, like presently, as you know, we are vaccinating with goat pox, and as on the last week, we have vaccinated nearly 8 crores of animals, and country presently having 97,000, little more than 97,000 active cases. And the good point is that last week we have only 788 new cases reported, and death was reduced significantly. And only issue is that death we are still reporting in 2 states, Karnataka and Maharashtra. Maharashtra last week reported 99 and Karnataka reported nearly 200 odd numbers of death. Why I'm referring this, that we need 2-3 points where we need deliberation to be done. In the field, when you have the infection, the symptom varied widely and there is a death also. So there is a reason, there is a scientific way to find out why there are certain cases death are more or involvement of the internal organs are more. Like particularly in Karnataka, Maharashtra, the animals, we are still showing there is some death and there is a classical symptoms of LSD. So already we have decided to formulate a team that I will come separately and send it to Maharashtra so that it can be investigated. Regarding vaccine, as you all know, already we have got vaccine, homologous vaccine, as Dr. Tripathi has elaborated on that. Different people have taken the technology, Bharat, IL, and even Maharashtra government have taken up the technology. Vaccine production already at the different stage of testing. Here, there are 2 issues has to be discussed and deliberated, which Dr. Tripathi also raised, that whether there is a chance of any kind of reversal of virulence. And if there is a chance, what is the chance? And if there is no chance, how do you say scientifically there is no chance of reversal of virulence? And what is the scientific basis or parameter by which we can determine the chance? So there should be a scientific parameter and a guideline, depends on which we can take a decision on that issue. That is a very important issue. Second thing is that there are many number of people and institutes who have gone into the sequencing of LSD genome, right from CSR and National Institute of Virology, Pune, and there are many other institutes, and some of those are already available in the government. What I was told that when you have a vaccine, that vaccine should have some deletion in the virulence gene. Though I am not a virologist, but being a geneticist, I understand that part, that when you have a deletion either by human intervention or by passaging through cell line or a non-natural host, you can make some deletion in the virulence gene. So you still retain the immunogenicity, but you take care of virulence and you develop a vaccine strain. Now, this body, what you are meeting, I would request all of you to go through it. Just a minute. Hello. एक बार उनको बोलो थोड़ी देर बाद मीटिंग में हूं मैं. So what I was trying to tell that, there is a need to discuss and deliberate whatever the available sequence available in the NCBI or other public portal, as well as the vaccine strain, and to demonstrate that the vaccine strain has got the required deletion in the genome, in the virulence gene, if it is required, and it is able to immunogenicity you can demonstrate, but it will not cause infection or reversal is not possible. That part has to be demonstrated and clearly discussed and debated. And if there is no deletion, in case I have not gone through the genome as such, then it is to be also deliberated upon that in this case, the vaccine has been developed and there is no such deletion is required. But these issues to be discussed and deliberated, and with a clear recommendation should come only on the basis of, I echo the word of Dr. Tripathi, on the scientific basis and based on the facts and figures. Third, how if we go for a survey monitoring, how we will be differentiate the animals which are vaccinated or got a natural infection of LSDB or got a natural infection of sheep pox or goat pox or whatever other pox viruses. So that we need to have a clear methodology to go for serosurveillance as well as seno-monitoring. And if we had some kind of a some kind of a national plan to develop, the department will look forward to have that. But for the LSDV, the now requirement is we should discuss and deliberate and debate with open mind the vaccine candidate which have been tested at the different level, their genome and all the genome available in the public domain, and see and demonstrate that there is a required number of deletion is already present in the genome of the vaccine strain. If it is present, it is causing this, and if it is not present, we should be able to tell that it is not required. I think much I can tell at this point of juncture. I have to go for another meeting, which will be chaired by our honorable minister. I wish I could be here personally and physically during the meeting, which I could not be. Gulati Sahab, I'll be coming on 7th to 9th. I think I'll be in Bangalore. And one day I will, or half a day, I will spend time with your institute and your people and that kind of time. And excuse me for this time. I wish a grand success of this particular technical deliberation, and the department will look forward very eagerly for the outcome and recommendation of this workshop. Thank you. Thank you very much. Jai Hind, Jai Bharat.
For sharing your valuable time with us and for emphasizing about the issues that need to be discussed during this workshop. So thank you so much, sir. May I request Dr. Divakar Hemantri, sir, Organizing Secretary, to take up the rest of the session. Thank you.
Good morning, everyone. Respected Speaker, Chairman, President IVS, Gulati sir, Director ICAR-NIVEDI, Dr. Bhatta sir, Director ICAR-NIVEDI, Sanya, Director Nishad, Dr. Pallab Chaudhary, Joint Director IVRI, Dr. P. Srinivas, Head SRS NDRI, and Dr. Khilari, and Dr. Naveen Kumar, and colleagues. So we have just completed the inauguration session, and we will be starting the second session of the workshop, which is basically the presentations by the experts in the field who have contributed to the LSD. And for this session, I request Dr. R.K. Singh sir to chair the session, and co-chair will be Dr. Nikhil Sanyal and Dr. E.R. Gulati. Rapporteurs will be Dr. Jagdish Hiremat and Dr. Shivsharanappa. And we have about 6 presentations in this, sir, by Dr. Gulati sir, Dr. Naveen Kumar, Dr. Khilari, Dr. Niranjan Mishra, Dr. Mukesh Mehta, and Dr. Amit Kumar. Sir, over to you.
Thank you, Dr. Nadir.
Sir, R.K. Singh sir, if you excuse me, can I leave? And I forgot to say good morning to you. I'm extremely sorry I could not see you there.
No issue. No issue.
Yeah. Sir, please allow me to leave this meeting.
Please, because you already have meetings, so you can go to.
Thank you very much, sir. Namaskar. Sabhi ko namaskar.
So, please, we have now 6 presentations for this time. And yeah, so we are late only by 10 minutes as per standard Indian time. I think it is not too late. And I know all the speakers who are there, they are all eminent speakers, and they will definitely deliver their presentations within time. So before I invite Dr. Gulati, I would request Dr. Hemantri to go for a brief introduction of us.
Yes, sir. Dr. Gulati is a graduate from CCS Haryana Agriculture University. He has more than 30 years of experience on diverse veterinary viruses in the area of immunology and biotechnology, including rotaviruses, herpes viruses, adenoviruses, Japanese encephalitis virus, equine influenza, and avian pneumoviruses. Dr. Gulati's major contribution including contribution include unraveling the mechanisms of incidence of neuropathogenic and latent equine herpes virus infections in horses, the extent of Japanese encephalitis incidence in horse and pig population in India, epidemiology of rotavirus associated diarrhea in animals, and their divergence. They must play a key role in the development of vaccines such as equine influenza virus vaccine, equine herpes virus vaccine. Its contribution towards the diagnostic include a kit for serodiagnosis of Japanese encephalitis in pigs. His laboratory at NRC HISAR was involved in testing more than 25,000 human samples during COVID-19. Gulati is the recipient of many awards, including fellow awards of NAVS, IVS, ISVIB, and he is also one of the editors of virus diseases and ISVIB journal Veterinary Immunology and Biotechnology. Dr. Gulati is currently serving as the director of ICAR-NIVEDI. Thank you.
Thank you, Dr. Mati. Now I invite Dr. Gulati. And both of us, we have been privileged because we had an opportunity to work together. So over to Dr. Gulati, please.
Thank you, sir. At the outset, I would like to thank ICAR, Department of Animal Husbandry, Dairying and Government of India, and Indian Virological Society for giving NIVEDI the opportunity to conduct this workshop at NIVEDI in both hybrid mode. And I would also like to thank all the dignitaries present online as well as physically in NIVEDI. And also I would like to thank all the dignitary speakers who have joined in this workshop, and they will be deliberating on different aspects of lumpy skin disease virus and lumpy skin disease. So thank you all, and I would like to have my presentation shared. Is this presentation visible to everyone? I hope so. So, sir, I have been given the topic to speak on spatio-temporal dynamics of lumpy skin disease in India. And my team, Dr. Reddy, Dr. Chanda, Dr. Himatri, have initiated work on epidemiology and risk analysis. Dr. Suresh and Dr. Govindraj, they are now working on informatics and economics of lumpy skin disease.
Dr. Gulati, your slides are not visible.
Okay, sir.
आपको दोबारा शेयर करिए इसका अनशेयर अनशेयर
Can you unshare and again share? Is it visible, sir, now? Sir, is the slide visible?
Now it is visible.
Okay, thank you, sir. So I will be talking on spatio-temporal dynamics of LSD in India. Can I have the next slide? So lumpy skin disease, as you know, has entered in India in 2019 and has been a devastating disease for the livestock sector and specifically the cattle in India. And the disease has caused more damage to the livestock, especially the cattle, during last 3 years than any other disease probably during last 1 decade. So when we talk of the lumpy skin disease, from epidemiology and outbreak investigation, first thing is what is the case definition of lumpy skin disease for all the vets doing the field investigation on lumpy skin disease. So this is what is the case definition that every veterinarian begin with when they go for disease investigation. So a suspected lumpy skin disease case, when a cattle has fever, lacrimation, nasal discharge, salivation, followed by eruptions on the skin and other parts of the body, then the nodules of this lumpy skin disease, unlike other diseases, are well circumscribed, round, slightly raised, firm, and painful. And confirmed, this will be suspected case of lumpy skin disease if we find such kind of symptom. So confirmed case will be when coupled with these clinical case definition, the case is confirmed by laboratory diagnosis, then it can be called as a confirmed case. So about the transmission of lumpy skin disease, it is transmitted primarily by vectors like by infected ticks, infected mosquitoes, infected flies, and from the animals from one cattle to another cattle, it is transmitted by these vectors, and also through iatrogenic routes when it is a short distance, when animals are in short distance. But for long distance, the virus or the infected vector has, or the infected animal has to be moving, crossing the border from one area to other. Infected animal or the infected insect vectors, they can also be transmitting the disease to long distance. So clinical symptoms, as I described, everybody knows, following the incubation of 4 to 7 days, there is fever, there is enlarged lymph nodes, and then typical nodular lesions, which are of the size 0.5 to 5 centimeter. And then in some cases, as we found in India, it is a systemic disease also causing pneumonia kind of symptom and infecting different organs. So typical with the disease, there are, besides skin nodules, there are typical gunshot wounds in respiratory system, focal pneumonic lesions on lungs, multiple focal necrotic foci on liver, elevated ulcerative necrotic lesions in abomazum and many other organs. So coming to the epidemiology of lumpy skin, this triad of the agent, the host factors, and the environmental factors play a critical role in the epidemiology of LSD. So the agent, as I said, is a Capri-Pox virus. It is the same virus of the same family, Poxviridae, where the other viruses we are talking about sheep-pox virus, goat-pox viruses they are. And this virus is a single serotype. Single serotype means the vaccine should be giving protection by any virus that we use for vaccination purpose. This virus remains in blood of the infected animal for 5 to 16 days, in the skin nodules up to 39 days, and in scabs of infected animals, virus can stay for several years. Even in semen, it has been detected for 20 to 40 days. Coming to the environmental factors, virus is pretty stable, but can be inactivated by heat, acidic and alkaline pH. Then amongst the environmental factor, rainfall is a critical area because this is a vector-borne disease. So following the rainfall, as in monsoon season, following monsoon season, the severity of the disease is more, and many vectors are playing a role in transmission of lumpy skin disease. More than 35 flies and 17 of different 19 ticks have been detected positive for LSD by PCR by the NIVEDI. Then there are many host factors that is affecting the outcome of the lumpy skin disease. One is that although both cattle and buffaloes are susceptible to the disease, but cattle is more severely affected and buffalo is relatively not that much affected. And then very young animals are more severely affected by the LSD. Then similarly, the crossbred animals are more severely affected, more mortality, more morbidity in the crossbred animals. This is about the triad of the lumpy skin disease. Then the key question that we need to understand in epidemiology of lumpy skin disease is how this spatio-temporal distribution of LSD is taking place, how it entered in India, where from it entered, whether all the states of the country are equally affected, if not, why they are not equally affected, whether all the districts within the state are equally affected, whether there is any seasonal pattern of occurrence of the disease. Then also we need to have some questions on the control strategy. What is the duration of immunity with natural infection and with vaccination? Then how long we should vaccinate and where to vaccinate? these are some of the questions we'll be trying to address in subsequent slides. Can I have the next slide? So this lumpy skin disease, this is the first slide where, as we say, this is sometime in March 19, it was in Russia. Then in July, it was entering in Israel, and then subsequently from Israel and Russia, it entered into China and India in August 2019, and subsequently from next. In 2021, it reached up to Sri Lanka, crossing all the India, and then subsequently it went to, in April 21 to Taiwan, Thailand, the Southeast Asian countries. May 21, it reached to Laos, and then June 21, it reached to Cambodia, and then August 21, it was reported from the Nepal. Probably it might have been previously in Nepal, but there were no reports. And then it has been in the entire Southeast Asia by September 21. So what we think that the disease has entered somewhere from China due to the movement of either infected animal, unrestricted movement of animals or vectors or through waters, and it has been moving from high altitudes to the lower altitude from China to via either directly from China or via passing through Nepal. Can I have the next slide? So previous. So this is the population which is at risk for lumpy skin disease in India. 192.5 million cattle, which are mainly in the countries, the central and northern India. Then buffalo population of 109.85 million. Next slide. So in India, the disease entered in year 2019, August, from somewhere from Odisha. It was first reported, although it might have entered from China, passing through different states, but it was officially reported in August 19. And then by 2021, it spread to the many states of the country, and year 22, we see the entire country red with the disease. But what we are finding that in the first wave, 2019-20, there was, although morbidity was there, but mortality was very low, and the disease form was only the dermatitis, the skin nodules were there. But in subsequent waves of second and third waves, the disease has been causing more mortality and more severe disease. Besides dermatitis, it was also the generalized and respiratory lesions were there, causing a lot of mortality. So this is the now current status of the LSD district map. It has spread to almost entire country, and many districts are reported to be affected with lumpy skin disease. And official data that we could get from different states is 29,26,039 cases as up to December 22. Next slide. So the most severely affected states due to lumpy skin disease include Rajasthan, the most severest of the disease, Karnataka, then Maharashtra, Punjab, Haryana, and MP also. Next, so the number of states affected is 24 we have reported so far. The reported cases which are reported to the DASD or the NIVEDI, there are 24 states reported. Number of districts affected so far are 335. So in these 335 districts, the population of bovine population which was at risk is 24 crore 45 lakh, and total population affected is 29 lakh 26,039. Next. So if we see the comparative year-wise distribution of LSD outbreak, attacks and death, the initial year 2019, it was very low. Subsequent, there were 641 attacks and no mortality probably in 19. Then 2020 and 21, the more attack rates and the mortality started rising. But 2022 has been very severe. We had more than 23 lakh attacks and more than 1,28,000 deaths reported during 22. So if we look at the seasonal or the month-wise distribution of lumpy skin disease, as I said earlier, the post-monsoon period when the disease vector activity is more from August till maybe November or December months. During all the 3 years, 2019, we had second month that I am reporting. Peak was somewhere in August and September 20. Then next year also 2020 peak was somewhere in August, September. 21 also, although we don't have much of the data of 21, but still it shows the peak was somewhere in August, September. And now 22 also we are finding peak in August, September, October, November, and December. So this is the period that this is the countrywide data. The disease is spreading like that, and that is the most critical period. So probably this information will help to choose us if we are going for annual booster vaccination. What is the right time to boost the animals? Next slide. So if we see at the month-wise distribution, as I say, January probably 1 or 2 states having the cases. Next, February more very few states. Then March, March, April. Next quickly. So if you come at August, September, almost entire states having the cases, more number of cases. Yeah, this is August, September picture. Next, October picture. So what I mean to say is that this disease having more severity in a particular season when there is vector abundance. Next, so this is this showing the disease dynamics. We just take a case study of the Karnataka state, because this is the state where disease is currently active. And this western districts and the eastern district, Bellary and Belagavi, this is the where we are now currently getting the highest mortality. And the disease has been severe in year 2022 only in the state of Karnataka. The first, if you see at the right side top picture, this shows the reproductive numbers or the curve, how this over time or not is changing. And there was one peak in the beginning in the August month. Then when this R naught goes above 1, that means transmission rate of the disease is very faster and it is affecting more number of animals. And then subsequently we are getting many different epidemic peaks of this R naught or the disease transmission. This probably this figure shows this is not the one location, this is the entire state representing different districts. So what the message this picture is giving, that if we had started serious mass scale interventions at the beginning of the disease, the subsequent peaks, although they are subsiding, but would not have been emerging. If we go to the rightmost corner, we find that this peak is again trying to emerge again. So this means that some more interventions at the state level for the control of lumpy skin disease have to be taken so that this peak does not further develop into higher peaks. So can I have the next slide? So coming finally to the economic losses, because it is one of the criteria, the economics where we based on which we allocate our resources and prioritization for the disease control programs. So LSD is causing many severe economic losses, either by directly or indirectly. By reduction in milk, it is causing severe reduction in milk up to 40%, 50% milk reduction, draft power reduction, decreased body condition, damage to height, abortion, infertility, and even deaths also. Then besides these direct losses, indirect losses are also being caused by the lumpy skin disease by way of restriction on movements of animal and animal trade restriction, losses to other stakeholders like dairy, feed industry, transportation industry, cost on exchequer in the term of treatment and prevention control measures, difficulty in delay in implementation of government scheme, nutrition, soil fertility, organic farming. So these are the indirect losses. Can I have the next slide? So we are trying to have the total impact of the lumpy skin disease to the country, but initially we started with a few states, Rajasthan, Karnataka, Gujarat. We have analyzed some of the data of this state of Rajasthan because it was the one state severely affected. If we look at the individual animal-wise loss due to lumpy skin disease. This table is showing the result. Milk loss in indigenous cattle 5,800 rupees. Because of lumpy skin disease, crossbred 7,400 rupees. Similarly for mortality and treatment cost. And if we work out that indigenous cattle, 27,770 rupees per animal cost because of lumpy skin disease. And for crossbred cattle, it is coming somewhere 49,600 rupees. This is based on our intensive planned survey of the state of Rajasthan that our team are doing. And if we look at the farm level economic losses in Rajasthan, if we take the farm size of 2 to 10 animals, which is generally there, if you take the average size of the farm, 5 animals, then the total loss per farm is coming to be somewhere 55,000 rupees, although range is bigger from 14,000 to even up to 2,12,960 rupees. So if we look at these figures, we can make projections based on the 16 lakh cases observed in the state of Rajasthan. Total loss due to LSD in Rajasthan might be more than 11,020 crore to the state of Rajasthan, livestock sector. So if we work out the cost of the lumpy skin disease to the entire country, that will be very, very enormous figure. Can I have the next slide? Next. So coming to the conclusion, what are the challenges in prevention and control? I will not be focusing on vaccination and other control strategies because subsequent speakers will be talking about, but just focusing on some of the challenges as Dr. Tripathi, our DD sir, and Dr. Mitra and Dr. R.K. Singh sir have already pointed out. When this disease came regarding vaccination and post-vaccination monitoring, there were certain challenges. We had the issues of availability of required number of doses of vaccines. Then we had a goatpox vaccine that is currently being done in the entire country. Then there were questions: which are better, homologous or heterologous vaccines? That will be the topic of discussion during subsequent speakers' address. Then there is a question: when should be the booster vaccination done? What should be the timing of vaccination? How many times we should be going for booster vaccinations? Then for LSD, if we with the current available serological assays, antibodies are detected maybe 30 days post-vaccination, after which titers gradually decline to undetectable level. Although besides antibody, cellular immunity is also playing immunity to lumpy skin disease. So it is expected that based on the current knowledge, that post-vaccination immunity is likely to persist for 2 to 3 years. But we do not know the exact duration of immunity by available vaccines, whether by the goatpox vaccine or the forthcoming homologous lumpy skin disease vaccine. What will be the duration of the immunity provided by these vaccines? Then there are challenges on the nationwide surveillance of the disease. Once, because now entire country is infected, but subsequently the disease will be coming down, then there will be need of continuous surveillance of the disease to detect active cases. Then there will be challenge to detect the post-vaccination status of the immunity by doing the serological surveillances. So we have to have these syndromic surveillances or the clinical surveillances of the disease, because it is very easy to diagnose based on the clinical signs. Then we need to have serological surveillance in place. So during the cases of disease outbreaks we are getting, and subsequently post-vaccination, we have to have the response studies also, where we also need to have the virological surveillance to be continued, so that we can detect active number of cases. If we need to think of the control or eradication of the disease, then also there is a routine vector surveillance. That part probably we are neglecting. We are not focusing on the vector surveillance. Then regarding vector control, we are not thinking on that. Insect control, both in the environment and also in the animal farms, we have to have the measures in place for vector control, anti-mosquito nets or spot-on repellents, and then eliminating vector breeding places as we go for in vaccine, malaria or dengue. We have to mission mode programs for stagnating water, removing slurry from the animal farms, manure clearance, drainage system in the animal farms. These are the areas which can help in vector control. Then finally, biosafety and biosecurity at the field level is one significant area which can help in controlling and eradicating the LSD. The restriction of the movement of the animals, especially the infected animals, then cleaning and disinfection of infected premises and the environment, then quarantines of the new animals, new entries into the herds. We need to have a certain quarantine period, and then finally we will be needing an exit policy or exit strategy to eliminate lumpy skin disease. So what the current information provides us that annual vaccination following this outbreak control, we have to go for at least 3 years of annual vaccination. So we need to be ready for the vaccination, booster vaccinations. We must be needing some campaigns like pulse polio campaign in the entire country. Before the susceptible period starts, we need to have at least for 3 years. Following that, we need to stop the vaccination if we want to eradicate, as I was talking with DDG sir and the Animal Husbandry Commissioner sir, that they would like to have something, elimination of lumpy skin disease latest by 2030. So we, following 3 years of continuous vaccination, we will be doing surveillance. So what our strategy will be that disease should not be there for 3 years after vaccination if we are doing only clinical surveillance. But if we are doing clinical surveillance, virological surveillance, serological surveillance, then at least 2 years there should not be demonstration of disease, so that we can attain the freedom from lumpy skin disease by maybe targeted by 2029 or 2030. Next slide. Next. So we need to have also the alternate strategies for when this outbreak is over, when this is, we will be getting, we expect that for following 2 years, next 2 years, because of vaccination, we will be getting sporadic cases. We need to have the strategies for controlling those sporadic cases of lumpy skin disease in country. And after 3 years of vaccination, we need to have stringent surveillance, border vaccination, blanket vaccination, zoning in, or ring vaccination, that kind of thing. We'll have to have that approach so that we work seriously and dedicatedly for getting the zero lumpy skin disease campaign. Next slide. So finally, we need to have, there is a need to have the strengthening of surveillance on lumpy skin disease, nationwide surveillance we need to have system. Then uniformity in data reporting. We need to develop some structure like we had in COVID-19, where cases were reported day-wise in real-time mode. So far, we are getting the reports of lumpy skin disease maybe weekly or monthly, but we need to have the reporting system strong, that we should take the advantage of COVID-19 experience for uniform data reporting in real-time mode. And then vaccination strategies we need to have the place. We should have the documentation of the animals, those who have been vaccinated. Because now the DST is running the ear tagging or the Pashu Aadhaar programs. We should be documenting which vaccines, which animals are vaccinated, which vaccine is being used, and then when is the time for the next booster vaccination. This documentation, so far, we are not working on that. We need to do that. Then we need to have uniformity in diagnosis of the disease, whether we need to diagnose the clinical cases, whether we need to have the DIVAs for differentiation of infected and vaccinated animals. And then we should have approved labs, countrywide approved network of approved laboratories which are authorized to test cases for lumpy skin disease. And then finally, there is need to develop a lumpy skin disease control action plan document. And this workshop is probably one of the steps toward developing that action plan. Thank you very much.
Thank you, Dr. Gulati, for an excellent presentation. Get it cheap now. And I think that Gulati has covered from spatio-temporal distribution up to way forward. So it's a very good presentation, and I think there has been clarity in the presentation. So people who have attended, now we are more knowledgeable about it. So how do you wish to go ahead? Do you want to take up the question now? If there are any
Couple of questions, and otherwise.
maybe few
In the panel discussion, we can have.
yeah. So maybe we can have 2-3 quick questions from the audience, online or offline.
No, we can have a discussion.
Yeah.
One has come.
Yes. Okay, now I think now we can go to the next speaker. We'll take up these questions later. Now we have Dr. Naveen Kumar from NCVTC ICAR-NIVEDI ISAR, and he will be presenting on vaccination and control strategies. I invite Dr. Mardi for a brief introduction.
Dr. Naveen Kumar is a well-known figure, and he works in NRC once. He has completed his BSc in 2000 and MS in 2002 from the College of Veterinary Sciences and PhD from Research Institute in Germany. And CCS at Haryana and a Spanish program. He served as a postdoc at Emory University, Atlanta, USA from 2006 to 2011, and worked on understanding the interactions of influenza virus with the host signaling pathway. He joined the Indian Council of Medical Research on 2011 as a senior scientist, and is currently serving as the principal scientist at NRC. It was particularly the National Center for Veterinary Diagnostic Courses. Dr. Kumar's research interests include virus-host interactions and identification of several key cellular kinases and epigenetic modifiers like MNK1, SCRCA, FEPS, AF4E, ALK5, P38, OC1, lethal transferases, etc., which are essential for virus replication but are dispensable for the host and hence may be targeted for antiviral.
Sorry, your slide is not visible. I mean, आप लोग अपना system ठीक करो.
He has been a key contributor in the development of India's first homogeneous live attenuated LHC vaccine. Besides, he has also developed one first indigenous vaccine to prevent coronaviruses in animals. Currently serving as associate editor of Virulence and a member of editorial board of several journals. His research works have been really published in many different journals, including PNAS USA. I invite Dr. Kumar.
Dr. Naveen, what do you see?
Slides are visible, sir.
Yes, visible.
Okay.
Good morning, everybody. Thanks to the organizers and our honorable DDG who have organized this important workshop on the. So I am from the National Center for Veterinary Type Culture. So we are a repository that is located at the National Research Center on Bitcoin ISAR. So I think much has already been said about the epidemiology control measures. So I'm just going to start about the vaccine. So you know the entered in 2019 in India. So we had no option because we don't have a homologous vaccine. So, but fortunately, we had a good folks vaccine technology. So, policymakers in our country, they authorized the use of the good folks vaccine. So, initially, it was the Hester Biosciences only, and then after in 2021,
the Indian Immunological also got the license to manufacture the goatpox vaccine. So because initially the goatpox vaccine was for the goat, and then there was a huge shortage of supply. But later on, as of now, I think the vaccine is abundance and no shortage of supply. So far, beside the vaccination, the one important aspect is to control the vector. But as you know, DGGs are also told, and other speakers, they told that we have a very poor understanding about the biology of this LSDV. So a lot of things we don't know. Even about the genome, we don't know a lot of the portion of the genome. We don't have any information what it codes for. So the transmission via vector is a mechanical, but actually we don't understand. And at a global level, there is very poor understanding about this transmission. Of course, is by vector, mechanical. So we need to undertake some of these studies in India. Right now, I don't believe anybody is undertaking such studies. I had some discussion with Dr. Manjunath. So he wants to go for it, and definitely we should have some projects on to understand which nature of the vectors, whether the virus undergoing biological change, and how to efficiently control the vectors. Next slide, please. So next, the goatpox vaccine, as I told you, it was authorized, but the extensive literature suggests that it's not as efficacious as the homologous vaccine. And of course, in our organized farms, like the NDRI, Gadwasu, Ludhiana, and IVRI, we have seen that the disease, natural disease appeared after a few months of goatpox vaccination. So at the international level, there is consensus that the heterologous vaccine, they provide the partial protection against LSDV in cattle and are not as efficacious as homologous LSDV vaccines. So for right now, we are using the goatpox vaccine. So we had in at the time of the COVID, thinking of initially just trying to isolate the virus, but then at that time it was realized that we must have a homologous vaccine. So I started adapting the virus. Next slide, please. Yes. So the story, starts from a request from the dean, the College of Veterinary Science at BITS Agriculture University, Ranchi. So in December 2019, DDG sir at that time, he was director at our institute. So we were actually looking to isolate the virus. We don't have, at that time, the idea of developing a vaccine. But I visited there and we collected the samples, and then I isolated the virus. Next slide, please. So I'm not going to go much detail about the virus isolation part, but that was published in the PLOS one. So then we when there was a COVID, and most of the labs they're almost closed, so we were actually adapting this virus. So virus was originally isolated in the primary goat kidney cells, and then we adapted it in the viro cells. So initially, it took about 10 days before to see something. But later on, the virus started replicating much faster. And at passage number P50, virus produced CP within 2 to 3 days, depending on the MY used. So at P50, we had we sequenced the virus at a complete genome, and we analyzed that virus has acquired sufficient, mutations and lost its virulence genes that we have seen in the sheeppox and goatpox vaccine strain. Next slide, please. So, these are the virus was, at the, completely sequenced at the wild type at level, passage level 10, 30, and 50. So when we compare the P50 virus with the wild type virus, so we were able to see that some of the virulence genes that are fragmented. The majority of the major mutations were observed in the calci-like protein and pyridinic protein, envelope phosphoprotein, DNA polymerase. Also, intriguingly, there is 12 base pair insertion in the GPECR region, which is present in all the vaccine strains, including the CIP and Gotopox strain. So the question that was raised by the Commissioner ICAR-NIVEDI, so I would like to clarify that the existing CIP and Gotopox strain, they have the major mutation is these proteins. So it's we believe that the virus, the necessary modifications or attenuation has already taken place at P50. Interestingly, in our strain, that there was a 801 base pair deletion in the inverted terminal repeat region. That 801 base pair deletion is not present in any of the other existing strain, vaccine strain, whether it's Cpox or Gotopox strain. Next, please. So, then if you want to make it a vaccine candidate, that the virus should go at appropriate titer. So we did the growth curve analysis, and you can see that virus grow about 10 to the power 70 CID50. That's good for making the vaccine. Next. So based on the genetic signature, that the fragmentation of the virulence gene, which has been also reported in other Mpox vaccine strains. Also some laboratory animal studies in the rabbit mice, although they are not natural hosts, but for safety aspect we did it. Then, with the help of the IVRI mutator, Dr. Amit, and we conducted the challenge experiment as per the OI protocol. So it suggests that you must vaccinate 10 animals. 2 animals should receive 10 times field dose and 8 should receive a single dose, and 5 animals should be controlled. So that experiment was conducted at IVRI mutator. Next slide, please. So the major problem with the existing vaccine, the live attenuated homologous LSD vaccine, is that up to 10 to 15% animals, they develop the local reaction or sometimes generalized reaction. So major problem of the vaccine is the safety. So so far out of the 10 animals, none of the animals they develop neither the generalized reaction nor the local reaction at site of injection. Occasionally, a few animals at a certain point they showed the fever, but we couldn't ascertain whether this is related to the vaccine. There was the viremia, we can say, but we couldn't isolate the virus. The viral genome was detected in the blood of the animals at 3 days post-vaccination, but virus could not be isolated. There was no shedding of the virus in fecal, nasal, and ocular routes that were periodically examined. So the animals, they were quite, remain apparently healthy. There was no anorexia, no swollen lymph node, no weakness that we observed in the naturally infected animals. And all the blood biochemical parameters were also all in normal range. Next, please. So if you see the individual animal performance, except the red boxes say that there is the fever which we were able to see in some of the vaccinated animals. Otherwise, in all the parameters, they were quite okay. There was no excretion, there were no lesions, there was no viremia. All those few animals, they showed the presence of the viral genome in blood. Next slide, please. So these the blood pictures, following vaccination at different days. So this all looks normal, So as compared to the control. So there's no unusual hematology biochemical parameters were observed. Next, please. So then, at day 30 post-vaccination, the animals were challenged. So before actually going to challenge the vaccinated animals, we want to ascertain that the hot virus, that is field virus, what we have, is able to produce the disease or not. So the virus was, the animals that were seronegative were challenged with the field strain, that the same strain which was used to make the vaccine. So virus was, the animals were challenged by intradermal as well as the intravenous route. And we were able to see, you can see the localized lesions, some generalized lesions, and also swollen lymph node, anorexia, and of course the fever. So this suggests that the hot virus, is produced the disease. But it's, in lumpy skin disease, if you do the experimental infection, so the outcome is variable. So all the animals, all the time, they do not develop the lesions or classical lesions of the LSD. So for us, we injected in 3 out of 2 animals, they develop the symptoms of the LSD. So this is, this is seems to be in order as per the OI literature. Next, please. So for challenge, 5 unvaccinated animals and 10 vaccinated animals, they were challenged by intravenous route. 0.25 ml intradermal at 4 places and intravenously 2 ml. Next, please. So for the efficacy of the challenge infection, the vaccinated animals, they did not show any inflammatory response at site of inoculation. No skin nodules at site of inoculation, no generalized nodules, no viremia, but some of the animals, they showed the fever. But as compared to the unvaccinated or control animals, they all develop local and sometimes generalized reactions and develop fever, viremia. I mean, all the parameters, of the clinical signs of LSD can be seen there in the unvaccinated animals. Next, please. So then, about the immunogenicity. So, I keep continue, receiving calls from several colleagues that how much is the antibody titer that is, we can consider that it is safe to protect the animals. But this organism is not like FMDV, where the antibody titer well correlates with the challenge data. So in our experimental trial, out of the 10 animals, 9 of them, they showed the antibody at day 30 post-vaccination. So you can say the seroconversion was 90%. We also conducted field trials, which data I'm just going to show you after this slide. So out of 26, about 26,000 animals, we have collected, analyzed about 650 serum samples. So seroconversion rate was 85%. But I believe this is a little at higher side because some of the animals, they have been exposed, to the natural infection in between. So we measured the cell-mediated immune response as well. So there was both experimental trials and field trials via the lymphocyte proliferation assay. So it appears that it induced a good cell-mediated immune response. And we also observed the interferon gamma response by the bovine interferon gamma kit. So the highest IF and gamma response is at 3 days post exposure. Next, please. So this is the individual animal performance following the challenge. So right panel is, the control animals. So these are 3 are the control animals. Red boxes indicates, either they develop the viremia, fever, or lesions. So you can see in the left panel, which are the vaccinated animals, all the animals, they are clean, except one animals at a few occasions it showed the fever. Otherwise, the control animals, they all developed the lesions, viremia, and the fever. Next slide, please. So then we, at a very high time, when these data, we were analyzing this data, then there were extensive outbreaks started, particularly in the Rajasthan and Gujarat. So then it was decided with the competent authorities that we must take some field studies. We must go to some gosalas and try to vaccinate and see the efficacy. So although till now we have vaccinated over 2 lakhs animals, but data I'm presenting is only about 27,000 animals. So this includes the 6 states: Jammu Kashmir, Rajasthan, Haryana, Uttar Pradesh, Delhi, and Punjab. So 26,940 animals were vaccinated. So all the animal details were taken before the vaccination, and it was under the observation of some field veterinarian or with the faculty members if is attached with the university. So if we regarding the species, so out of the 27,000 animals, most of them were cattle. It also includes 413 buffaloes, and it also includes pregnant cattle and buffaloes that were about 2,889 in numbers. And of course, we also had a record of the lactating cattle and buffalo that was over 10,000. Next, please. So this was actually the performa which we circulated to the farm manager or the competent authorities, and we seek the information about total number of animals, whether there is any swelling at site of injection, generalized injection, daily milk production, reduced milk production if any, and if there is reduction of milk production, how long and when it was resumed, if there is any abortion. Appearance of the disease, because the extensive outbreaks were going, so it's likely that some animals may expose before development of a complete immunity. So we recorded the disease before completion of 3 weeks of vaccination or after 3 weeks of vaccination. Next slide, please. So one of if you want to develop it as a product, then of course this must be safe and should not have any public health hazard. So the vaccinated animals in field, they were subjected to for detection of the virus in the milk. So 2 farms at day 3. In one farm we collected the milk at day 3 and 7 post vaccination, and another farm day 7 and 14. So none of the animal they showed the presence of the vaccine virus in the milk. Although in the natural infection, some of the animal they excrete the virus in the milk. So we were lucky that with the help of Dr. Gulati, there is one organized farm in the ICAR, and he approached me for vaccination the bulls. So he supplied us the semen of the vaccinated bulls at day 10 post vaccination, and we tested the semen on all the animals bulls that were tested were found negative for the vaccine virus. Next, please. So then this is the overall picture of the field trials. So as I mentioned in the beginning, the major problem of the existing the vaccine that is based on the needling strain used in Africa and some other countries. So it produces the needling response. Needling response means some animal they develop local lesion and sometimes generalized reaction, fever, and reduced milk yield. So far, we have vaccinated over 2 lakhs animals. So if I only go for these data about 27,000 animal data, 5 we have seen a local reaction that appeared at day 2 post vaccination and that subsided within 5 days. So this is actually negligible as compared to the needling strain-based vaccine, which produce up to 15% of the animals they show the needling response. So there was skin nodules. Of course, there was no skin nodules in any of the 27,000 animals. There was no generalized skin nodules. There was no viremia, although we only examined 62 animals. Fever, there was no fever observed, although only few of the farm in the university setup they were able to examine the fever. The vaccine virus excretion in nasal, fecal, or oral and fecal route, we examined 62 animals in the field, so we didn't see any vaccine virus excretion.
Likewise, there was no excretion in milk and semen. Milk production in out of 102 farms, two of the farms, 4 farms, there a small drop in milk reduction, but that was not significant. If we talk about the vaccine effectiveness, so out of 17,000 animals, 14 animals, they develop the disease after completion of 21 days. But that may, most of them out of these 14 animals, 9 were cows. But that may be due to the, in cows, maybe some non-specific interference of the maternal antibodies, and maybe they were not vaccinated properly. So at vaccine efficacy, we can say at this stage or rather effectiveness is greater than 99%. Next slide, please. I'm just about to complete. Next slide, please. So in conclusion, the based on the experimental field trials, so vaccine is safe, is better than the existing needling-based vaccines, and it provides protection, which we have seen in experimental and field trials. Next slide, please. So then, once if you know at some of the farms we had a problem, then the disease appeared before completion of 3 weeks of vaccination, and it is believed that the vaccine animals had developed immunity. So there were 16 farms where there was a problem. The disease appeared day 2 post vaccination to day 10 post vaccination. So in that case, it is necessary that we must differentiate the vaccine and field strain. But even in that case, the vaccine has a beneficial effect, because if we consider that the data that is being submitted by the government of Rajasthan, the overall morbidity rate and case fatality rate in Rajasthan was 11.7 and 4.8%. But in our case, in animals that succumb to infection early post vaccination, the morbidity was less and the case fatality was less, suggesting that it's beneficial effect in the outbreak situation if we are vaccinating the animals. Next slide, please. Next slide, please. So we have developed a test to differentiate the vaccine and field strain. As I told you that our strain has a 801 deletion. So that can be consistently observed at P50, P60, and P70 passage level, and also it's consistently 970 base pair fragment is in the field strain of 2019, 2020, or 2022. Next slide, please. We also developed to quantify the proportion of the field and vaccine strain by a real-time PCR. Next slide, please. So one of the question, is we keep continuous raised that 2022 there was more mortality, maybe virus has changed. Some guys in the ICAR-NIVEDI published some data that virus has some mutations, but we conducted a cross-neutralization assay from various viruses that were originated in 2019. That was vaccine was made 2021 from Banswara, Rajasthan, 2022 from Jalore, and also a camel isolate in 2022. So we have from our experimental trial, the sera derived from vaccination or from the infection. So they equally neutralize the vaccine or naked or infected sera derived from the 2019 virus. They equally neutralize all these viruses. This suggests that the vaccine, which is made by using the 2019 strain, is capable of neutralizing all these strains and is going to be effective. Next slide, please. Then again, another question is that if we have used the vaccine, that we must differentiate the vaccinated and infected animals. So as I told you that in our strain, there is a unique deletion, 801 base pair deletion in the inverted terminal repeat region, which is present at both sides of the genome. So we, after bioinformatic analysis, we observed that this is a homolog of myxoma virus M004. So it codes for the host immunomodulatory protein and modulates apoptosis and inflammatory response to virus infection. And most importantly, if you want to develop a DIVA, this gene, the protein which is codes, it induces antibody response in host. Although we don't know in LSD, but because based on the homology of the myxoma virus. So next slide, please. So this is the paper which suggests that this region which codes for this the immunomodulatory protein is immunogenic. So what we are going to do is that if the animal is infected, so it has the ITR region intact, and it's going to produce the antibodies against the viropine, that is the portion which is deleted, LSD3 and 154. But if he's vaccinated, because this virokine is absent in terms of the 801 base pair deletion. So if you express a recombinant protein of this virokine and try to react, allow the sera to react, that means the protein will only react to the infected one, but not the vaccinated one. Next slide, please. So this vaccine has been released in August 2022. Next slide, please. And the technology has been transferred to at least 3 of the firms, and BioVet has taken a lead and is already prepared 3 test batches and testing for IVRI. And we are expecting the product to be available soon in the market. Next. So there are a lot of peoples, there are a lot of field veterinarians and the other peoples which I can't acknowledge, but these are major contributors who had helped us this vaccine. Thank you very much.
Thank you, Dr. Giri, for an excellent presentation, and I think a lot of data has already been generated. One point what you said about post-vaccination, there was some infection. So these children, they were already in incubation period. So it is not adverse impact of the vaccine.
No, sir, no, sir. So there were 2 groups.
So try to understand. Those animals where you got seasons on day 2, means they were already in incubation period. It is not part of the vaccine.
Yes, yes. Yes, sir.
But as you said, because we also had done some work in this area in sheep parks earlier, I had the phase of an outbreak which happened in Rajasthan. I think that's what I remember. So initially, what will happen once their animals are in incubation period, you do not know. You vaccinate. So apparently, there will be some transient immunosuppression by any live virus vaccine. And it will seem that there is increase in the disease symptoms. But then ultimately, a total outcome, a cumulative outcome will always be beneficial if you are vaccinating the animals at the phase of an outbreak. So that's one point. Well done. Second point, why did you put question mark? If antibodies are not coming, you put cross so that it is understood. Question mark, anybody will say that it is not concluded because that the 800 base pair, 801 base pair already deleted. So I was talking to Dr. Gulati that this is a good signature for your vaccine.
Yes, yes.
And I'm happy that you have also developed the test for it. And then you have also developed real-time PCR. Yes. Now, next step for you would be to develop serological assay so that you can, at mass scale, this can be done. I'm very happy. Very good data now you have generated. But keep on generating data. Yes, thank you. And second point is like a large number of animals vaccinated at 3-4 scenarios I told. Try to get some serum samples about duration of immunization. Yeah.
We are analyzing.
That also keep in your radar.
We already get more than 3,000.
Okay. Thank you. So, please, we are already late, so I go to the next speaker. But before inviting Dr. Kilari, I would request Dr. Madi for brief introduction of Dr. Kilari.
Thank you, sir. Many of you must be knowing Dr. Kilari. He has been with the industry for quite a long time. I mean, in his almost 29 years. And of these 29 years, I think he has been with 23 years. I've been in the MNC environment. So whoever is in the academics definitely know him. And he is currently working as executive director of BioVet Private Limited, Homologous. And he is a veterinarian who do not know him. With a postgraduate degree in veterinary biotechnology from ICAR-NIVEDI. He has a rich experience of working in the industry. And he started his career as a research scientist at Bharati Agro Industry Foundation, founded by Gandhi and late Shri Manibai Desai. During his tenure, he extensively led the R&D projects on vaccines, on all animal species and its commercialization, initiated and managed several product development projects aligned with regulatory compliance requirements, collaborated closely with functional teams such as marketing, manufacturing, quality chemical, quality assurance, regulatory, the global and international environment to achieve business. Prior to his current position, Dr. Kilari has worked as a Chief Operating Officer BioVet, then Director at the MSD, Animal Health Innovation Singapore, and many other positions in Intravet India Private Limited and Hex Russell. In the sense, he has worked in multiple companies and almost sitting in the same place for most of the time. So I request her to invite him. Thank you.
So now I invite Dr. Hilary. Police, we are already late by 34 minutes. So I request all the speakers to save some time so that your lunch is not late.
Thank you, sir. Thank you very much, sir, for having this opportunity to be here, Nivedi. And thanks for Dr. Gulati and Nivedi and Apuriti sir, ICAR. Thanks, RK Singh sir. I think you are the right person to be here to be in this chair this session, and we have opportunity to talk about it. So those scientific angles have been mainly discussed by the field. So,
Microphone is there. Oh, like other audience people.
who can sit in the
Microphone is there.
You can use that also.
Why is it low?
Hello, is it I am audible now? Hello? Yeah. So now what of course, in my side, it is my colleague, Dr. YKM Reddy also. You all know the Professor YKM Reddy is the virologist and he works on viruses. And he is the one associated for the vaccine development technology transfer. So my part is much more easy because the previous that is the most critical part, epidemiology part and vaccine development and control therapy part, what I have made it. After getting this vaccine, yes, we had a kind of challenger. And what we can, before sharing the actual that one, maybe I would like to give the few minutes for the introduction of the organization also, so that industry perspective, most of the people have the better view on it. Please, next slide, please. We have the two campuses. The BioVet is basically it's at Malur, Karnataka. It is around 40 kilometers from this place. And it was started around 15 years ago. And last in 2021, earlier, Dr. Ahmadri was mentioning, yes, same place I was working for almost 25 years. So that place, it was belongs to MSD earlier, Intervet and next BioVet. Of course, it is taken over by the BioVet now. It is part of the BioVet now. So now that is how we have two facilities, vaccine manufacturing. This is all you know, that one is the BSL-3 facilities, that is which is required for foot and mouth disease vaccine. That is the major thing. So we have more than 500 employees who are working in the both places. Next place. And it is strategically is a very good thing that one we have the opportunity to work on the two different locations, BSL-3. And we have you all know that one foot and mouth disease is the trivalent vaccine. Initially, we had only one facility at Malur, one BSL production suite. So that we have to when you are manufacturing a vaccine, you have to make every time 1 serotype. So you have to make over a period 3 serotypes, then you have to make the vaccination. Dr. Yadla, you all know that one BioVet, they owned by Dr. Yadla, executive chairman. He thought about well in advance and he created two more production suite, BSL-3 facility. So that is how it became that new production facility became handy when COVID issue happened. When Covaxin was in need of that one in the large quantity. We used this new BSL-3 production suites, two more production suites, brand new, that were very handy for manufacturing the COVID vaccine. Now we have that one, we have changed it to the manufacturing foot and mouth disease. So that is advantage we are having. Parallelly, we can make all the 3 serotypes. We can get the vaccine at the end of the that is the advantage we have it. Next one, please. And this is the typically the layout. I mean, most of the people may be not familiar with the BSL-3, what it is mean that one. It is the typically on the first floor, if you see the ground level, it is the big cell fermenters and all will be there. And on the top portion, it will be there, the air handling and all the control systems, both cooling and distribution system. And bottom level, you have the drainage system. So that any escape and whatever the effluent and all, if it is getting escaped, you can contain very well. So that is the typically the BSL-3 facility. And if you see the left side is the leftmost is the expanded facility. That is how where we have the very big production facility of 10,000 liters. We are the largest manufacturer actually. Next, please. This is the, you can see the left side is the kind of automation with the transfer lines and fermenter reactors, everything at fingertip. And there's a lot of controls. What does it mean is we have more control on the manufacturing process. Nobody can tamper, or the kind of technician or the operator or who is doing. So whatever is doing, we can document, we can record the documents, live activities. If something go wrong, we can easily able to trace it back. Next, please. Similarly, we have the biocontainment for animal facility also. Next, please. And in addition to the BSL-3 for the manufacturing the inactivated or vaccines like we are also having for live vaccines for is a lyophilizer. So this is a huge capacity. At a time, we can have 100,000 vials per batch. What does it mean is per vial, if it is 100 doses, we can have the 10 million doses at a batch. So that is the Dr. Yada's vision actually. Nobody thought about this one, but even I could have not thought about. But he thought about that one. Okay, let us have the bigger capacity for any pandemic kind of situation. We are prepared. That is the way it is for the preparation is going. So this is the way we are using currently LSD vaccine. Next, please. So another facility I was talking to just yeah. So that's why Dr. Yella, Dr. Krishna Yella, you all know that one by this time. He is the founder of Bharat Biotech. He is the one of the NRI who returned back to India after his doctorate in University of Wisconsin, and he was a faculty. He was settled there, but he was doing very well. When they at the time when the government of AP, when they were inviting the people to come back and set up the industry in Genome Valley in Hyderabad, you all know that one, Genome Valley. And when they were invited, so he is the one of the first person, he came back and set up the Bharat Biotech at Hyderabad. So that is how he started 25 years ago. Later, he started Genome Valley as revolutionary, and many biotech companies has come up. And he is fully dedicated, and he is one of the visionary. He wants India to be more in the visible in the global. So that is the way he is looking at. Even this facility where I was working, I started my career in Bay in 92, when I was one of the last person interviewed by the Gandhi and Dr. Manubhai Desai. And that was a very basic rural development organization, particularly for veterinary. I am the one of the person, I didn't have any probation. There is a respect for the veterinaries in Bay. The moment day of joining is the confirmation. So that is the respect he used to give to the veterinarian. So probably I never had any probation in my career. So Dr. Yella is basically is agriculture graduate. So he graduated from Akola, the Maharashtra, and he did his masters from the Bangalore, the agriculture campus. Then he was went for the USA for his doctorate. So he's basically molecular biologist. So that is where he come across about hepatitis those days, hepatitis issue and all was going on. Then he is already having the molecular biology knowledge. So then he thought of why can't we make the vaccine for the country? That is how he came back and he started working on the hepatitis. That is how he started. But later went on for several vaccines. One of the few contributions to him is, to name it, it is one of the rotavirus vaccine, one of the vaccine which has been developed together with Dr. Bond from AIMS. You all know that was DBT secretary. So that is the one of the thing. Even before COVID, actually, Dr. Yadav was prepared Zika. He was expecting Zika is going to have some impact. He was preparing. Most of the people not know about this one. Zika vaccine, he was ready. In case if it was spreading and if something happened. So he was ready at that time. Bharat Biotech was ready to have that vaccine. So then he has not demotivated, but still when COVID issue was there, he took lead. So that is how we could come out with the COVID vaccine, and we all got that benefit. So now even the facility, Pune, somehow what happened, this facility was set up in 1975. It is a duplicate of the IVR Bangalore. Both facilities started same time, 2 projects from Darida. So this facility after, because Bay was a rural development, they don't want to be an industry kind of thing. After demise of Manubhai Desai, they decided at that time Hext was looking for the manufacturing for foot and mouth disease vaccine. So then this facility was transferred to them. But yeah, so later internet and sharing flow and MSD, there was transition of the place, but they couldn't sustain for the single reason is industry, anything you need to sustain, it has to generate revenue. So that was the model actually that was not able to sustain because of the turnover, the multinationals, they expect the quality, everything maintain the global level. They also expect the price as per the investment. So because of tender and all, they couldn't sustain actually. So that is how this facility was made. Dr. Krishna came to the risk of this facility when MSD decided to close down this one. Next, please. So these are facilities, almost we have the 2 sites, same as good as the facility, but it is a smaller capacity. Next, please. Next, please. So these are the annual facilities, we have that one. See, to summarize, so this is the largest BSL-3 facility. We can make the vaccine for current ongoing control program. We are fully geared up, almost 50% of the capacity of requirement for up to 500 million doses we can supply. Similarly, for LSDV, so we can make the vaccines. Actually, it is as good as billion if we are exclusively making one vaccine. So that is the capacity we are having. Okay. So next, similarly, we have animal facilities also. Next, please. So these are the vaccines. So coming to the proper, so this is the LSD vaccine. You all know that when we got the technology from NRCE, equine, Dr. Naveen, what he has already briefed. So this is the vaccine. We got it somewhere in the September, and we came back, and our team, led by Professor YKM, they had been to NRC, got the technology transfer. And so next, please. After coming back, we do have several challenges. We had several challenges, still we have it. Next, please. I have put some of the points here, keeping in that view. Most of the people and the institutions and all, they are interested to develop some new vaccines also. I mean, that is the reason. There are certain things to be kept in mind to make it the industry tomorrow to be the kind of not to repeat or not to duplicate the reinvent the wheel. Because if certain things is taken care at the inventor level, okay. So this is the something is really required, particularly cell and virus culture is very required. Most of the cell lines what we are using, I'm not sure whether it is really authenticated or it can be used for the commercial vaccine manufacturing. Because that is the first break. See, as long as you at research institutions you are working, nobody will pursue. The moment it comes to industry, we are accountable. Tomorrow anybody can put on the file, anybody can sue us. So that is the risk we are having it. So that is certain things we have to be very sure. And also one of the thing is most of the time we don't look into details of the external agents, the cell lines what we are having. Because what happened, particularly when you are inventing the new viruses, when you are adopting new viruses, there is a big chance that one the previous viruses or anything, it can come for anything, maybe from serum, maybe from trypsin, maybe cell line itself, or while handling also some of the human viruses. So these are the certain things we face challenge when we get the vaccine strain or the cell lines from the institution. Those are the angles we look into that one. So that is the time-taking process for us. In the same LSDV case, we went thoroughly. Of course, cell culture, we already said that one we are having very good cell line, which was already screened and free from exchange agent. We used that cell line to save the time. Because when the technology was transferred, we all know that one, we need a vaccine for emergency. And always I was told when I was in Delhi, you are given the Covaxin, so Dr. Yelda's on the pressure on him, you are given human vaccines, but you are not doing anything for the veterinary. So that was the why can't you do it? So that is how the LSD, when you took it, someone is the day one onwards is following. Even till today, you know that one I am here in this meeting, what I am going to speak. Okay. So now that is how he is also taking interest. There is emergency in the country, we need to address. That is the way he is looking at it. And these are the certain things we have to take it. Okay. Next, please. Now, how to, once we have the seed is okay, so how to make the drug substance? What does the drug substance means? It is a virus actually. In case of live virus, because it's already attenuated, this is directly you are going to inject to the animal. We are not going to do further any manipulation. Because that has to be, as the Dr. Naveen already told that one, that 50% there is a change, attenuation and all that one. So there is the reasons why it has happened. One of the challenge for us is how to scale up. When you are doing the small tissue culture flask and all, it is making the small amount of that one. For us, we have to suppose 100,000 vials I have to make. So I need at least 100-liter virus. So how to get this one into 100-liter virus? That is the main challenge what we had at that time. So we have different scale of operations. As good as conventional cell culture today, there is a self-active system. So that is something we had an advantage. That is a system we also use for making the seed cells for the viro cells of the SARS virus. Same thing became handy for us to scale up. Second one is even also we optimize the technology like that one microcarrier. This is not something widely used in veterinary, only in the human vaccines, because it's very expensive. Getting microcarrier is so difficult. So we have adopted that technology also for growing the virus so that you can scale up the volume faster. Okay. And most crucial thing what we had is the stabilization of the virus. That is the most critical part, because when you are going for the further lyophilization and all, we have almost worked at 70 different formulations actually. That was the biggest challenge we had, because it's time-taking process. Not only there is a different combinations of the salts you have to use, carbohydrates, you have to use the proteins, different things you have to be there to stabilize the virus. So that is what something we have narrowed down to the single stabilizer, which is the better one. Same thing, lyophilization cycles, at least 12 to 15 cycles we have optimized. That is how we have to able to optimize the freeze-drying cycle. Next, please. And getting the GMP system in place, that is the one thing actually, because the moment you are manufacturing at institute, nobody person you, but the moment we are making all this FDA, then DCGI, drug control office, everybody comes over there, they look into minute details. For example, I'll tell you, when you are entering into the manufacturing facility, what is the dress code, how the dress you are wearing that one. So then how you are disposing that one. Because what is the reason this? The process what we follow, or we are ensuring the quality up to end. That is the way they look at it. So historically, gone are the days, lot of manual operations were allowed, but now they are so stringent. So they are meticulous, they are very knowledgeable. I mean, that's what I'm telling. I really appreciate drug inspectors, what they have seen historically, what they are today. They are thoroughly trained with the WHO and all everywhere. Okay. So now after making the vaccine, so we have done that. These are the studies we have conducted. At least two things I want to mention most often being discussed is the safety. And one of the safety, I could really acknowledge the Naveen, what his observations, including the feed. Recently, I was there, one of the global meeting for LSDB at Dubai, wherein my ex-organization, MSD, has presented that the vaccine, what they are using for the nettling strain. After vaccination, when they show the picture of the animal, the entire body, there are micronodules. So that is something which is not there with this particular vaccine strain after vaccination. That is called nettling effect, what you was telling. So that is one of the best part, because the India, the moment if it is that one, nobody will accept. I mean, the farmer himself, normal, no need to be knowledgeable. That physical reaction itself, they will immediately, there is small nodules. Of course, they subside after a while, but in India it's not so easy, because we all know we are facing the FMD vaccination challenge. After 2 days of milk reduction is not acceptable. So many people resist the vaccination. So that is the one part safety I can assure. We also vaccinated, maybe Dr. Vaike can speak more on that one. So similarly, challenge test, what he has done, that one, similarly, the same study, we have reproduced that one, because we manufactured our other side. So we have to demonstrate the safety and everything. Most often was talk about the reversion to virulence. That is something which was widely was talking when before going for the technology. Even that was the question every one of us having it. So it has been vaccinated the animals, and then what does it means is after vaccination, so you have to take the sample from the animal, then you have to passage again in the animal. So that is called the back passages. So you have to do the 5 times. Then whether it is reverting to the virulent virus or any LSD related symptoms, because we have taken the attenuation in the cell culture, because away from the normal host, there is less likely it revert, but there is more chances it will revert back if we take it back to the natural host. So that is something is required. So that is the study. 5 back passages has been demonstrated. So already this has been full study has conducted. I think we also discussed with Dr. Naveen also this protocol everything. So that after 5th passage also there is no reversion to village. So that much we can assure you this one. And another study we are going further ahead and we are doing one more study. It is in progress. Very soon we'll be going for challenge. Heterologous challenge. What does it mean is you are vaccinating the current vaccine. We are all know that one goatpox vaccine we are using it. So we are vaccinating the cattle with goatpox vaccine, and we are going to do that is going to be challenged with LSD virus, the challenge virus. So that is in progress now. Very soon, maybe we will going further. We'll have the more information. I understood so far it was not done. That's what my earlier understanding. The goatpox vaccination followed by the LSD challenge. Nobody has done in the country.
I am not in the knowledge that.
So that's what I understood. That's what. So that is the reason I included this study in our proposal. Though it is not mandatory for us, but eager to know what it is that. Okay. So that result also very soon we'll be having it. And right now.
It has been done, but not in India. Abroad it is done, not in India.
Yeah. So now, so abroad, yes, so Dubai, there was some presentations where they said that one, the protection was limited. That's what they also showed. Okay. So now, right now, testing is going on at IVRI. Maybe it is in the final stage. They finished all in vitro testing. So they were also very excited after looking at the virus titers and all. So they are also safety completed. We had to hear about the potency test. And we also parallelly sent samples to Naveen also, though I have not mentioned here, because parallelly testing. So he already tested those vaccine batches, 3 vaccine batches. So recently there is also one more requirement. We also have to send the sample to the Bagpat, the other laboratory. So we are waiting for their confirmation to submit the samples. Next, please. So major thing is, apart from the production, okay, we are ready. Production is in our control. We can gear up whatever required. But what is that one is having the regulatory approvals. There are several bodies actually. So we need to have it. One of the positive this thing is that amount of data generated by Dr. Naveen, including field data, is enormous. Your company like for us to generate, it takes a lot of time. The only thing is limitation is when we were going with that data with the drug authorities, so they say that one. Whether you have these protocols are approved with us, whether you have the product what you are using in the field, whether it is manufactured under GMP condition. Historically, they accepted like PPR. So when we are doing the technology transfer from IVRI, we didn't have much resistance at that time. But now they are looking into more into those details. So those are the things. If it is we are really challenging, of course, we are trying to address. We said that one will be doing concurrently once we have the approval and all. And we already submitted all the approvals as process has been finished. I mean, we have submitted from our side. So we are waiting for final approval from the drug controller, and already inspection also facility inspection is over. Next, please. So this is the status, and we are waiting for eagerly. The moment we have the approval, as I said that one, we are ready to make the vaccine. We are waiting for the NOC from the ministry and the manufacturing license approval from the drug controller. So we are at the final stages, and we are thoroughly prepared. One of the challenge we had is the scientific people who can make the very few people worked on the pox virus. So a lot of people we have to train, and that's what the something. Thank you very much. Thank you.
I think this is good one before we are here. But most of the time we are not aware as a young scientist what is happening in the industry. And now we also know that industry is a very important role. Once we talk about having any national animal disease control program, or even otherwise, then you need the vaccines. So now, who is going to make it? Earlier, there were state biological production units, but now majority of them they are not working, except few like Maharashtra and others. Yeah, but then they can, even if they are able to meet their own requirement of the state, that's good enough. But for national level, we really need such kind of facilities. Your participation in this will also help us, but as regarded by DDG and also Animal Husbandry Commissioner, and so reiterated by Dr. Gulati, that maybe this workshop, today's workshop, is a priming workshop towards developing an LSDB National Control Action Plan. So there will be go for that action plan. Then how to meet the vaccine requirement of the country in terms of LSD. That also has to come in the action plan. So I thank you for your participation in the program. Dr.
Commission, I would like to share a few sentences about technology.
We are already delayed, so be very quick. Be very quick.
Actually, thank you for the opportunity given. And, congratulations to NCVTC for coming out with such a wonderful technology. Our team is actually spellbound. The way the technology was transferred, 4 areas are covered: virus propagation, virus authentication, safety, efficacy, data exploration, field data. Very well explained at NCVTC. Then we came back with the study and the technology. We got point to log extra only, the TCAD50, and 4 genes are authenticated, and including deletion. That concern was also addressed, deletion gene. And also sequence is authenticated, full genome sequence. And then we completed safety data with 3 batches. Animals are fine. We have not come across any adverse effects. Reversal to virulence also we have completed. 5 passages, there is no reversal to virulence. And efficacy also we have completed. Data is ready. Once regulatory clearance obtained, it will be made public. That is our experience. Actually, we are very much spellbound with.
Thank you, Dr. G, for illuminating us, because one of the question was also about attribution of the by age, so that is also added. So now slowly and slowly data is coming, and this is what it has been required in our previous discussion, that we need to have sufficient data. And you cannot silence people until you have this. You need to generate a lot of data. When we developed our classical swine fever vaccine, our own people at ICAR, they took, I think, more than 1.5 years. That every meeting they would say that you need to do this, you need to do this, you need to do 3 meetings it happened. Why I was very patient? Because when any product goes outside, you must be very foolproof. So generation of data is very, very important. I am happy that it is now at the right path. So please we move on to the next presenter. Before inviting Dr. Niranjan, I would request Dr. Imadi for a very brief introduction.
Thank you, sir. Now we are starting with the online speakers. Dr. Mishra will be joining from Bhopal. Dr. Mishra is a virologist with R&D and teaching experience on exotic, emerging, and re-emerging animal viral diseases. He presently works at the Principal Scientist at ICAR-NIVEDI. His major scientific contributions include work on ruminant pestiviruses like DBD1, 2, and COVID viruses, border disease virus, West Nile virus, infection in wild birds, LSD, and bovine leukemia virus in cattle in India. He has also been a key contributor to the action plan for minimum standards in bovine Trojan semen production and biosafety in cattle food semen station, and on diagnosis and control of LSD in India, including the molecular diagnostic protocols. He currently serves as one of the editors of animal virology in the journal Virus Disease. He has been a PI of several externally funded institute funded projects, and many projects on private and PPP mode. And his contribution on exotic diseases have earned him high accolades, including Hariom Ashram Trust Award. During his 27 years of career, Dr. Mishra has guided 6 PhD and 2 MSc students, and has published many research publications in national and international journals. So, over to you.
Thank you, Dr. Madre. So, Dr. Mishra, we are already late by about 44 minutes. So, I request to save some time, because your topic is on diagnosis, and until unless you really go for very rapid and quick diagnosis, then you cannot control the disease. So you need to be a bit rapid on your presentation also. Dr. Mishra, over to you.
Thank you, sir. Thank you, Dr. Timadri. Am I audible?
Yes, yes, very much.
Yeah. So respected chairman, Dr. R.K. Singh, and respected DTG Animal Science, and the co-chairman, panelists, and all other dignitaries present. And so good afternoon, everybody. So Dr. Himadri, can you share just the screen?
It is shared.
No, it is not visible now.
एक बार फिर से करिए।
Is it visible, sir?
Yeah, now it is visible.
Okay.
So next one, next. Please go ahead. Yeah. So I'll just, as Dr. R.K. Singh has told, I'll just give a brief about the disease and this status as well as the diagnosis, and mostly focus on the what are the diagnosis techniques are now currently, what is our strategies as for the Government of India guidelines. So this already it has been discussed by several speakers. It was a very old disease, but it is expanded. Geographic expansion has taken place in last decade. Enormous expansion to different geographical areas. Next one, please. So this was first restricted. It was historically restricted to the African countries. After that, it went to Middle East, then followed by the Eastern Europe, and then it came to Balkanic countries. Then again, the disease appeared in China, India, and Bangladesh simultaneously. Next one, please. So as you see in 2019, so most of it expanded into East and Southeast Asia. South Asia for first time. So the disease occurred first in June 2019 in June, China, Bangladesh in July, and in India August. Almost simultaneously, the disease occurred in 3 countries. So initially we thought that this virus might have entered through China or some neighboring countries. So, but afterwards, during our genetic characterization studies of the whole 2019 outbreaks, viruses from 19 outbreaks. So we clearly showed that this is not the virus which has come from China. This is a different virus, which is an ancestral Kenyan-like virus. The virus which is circulating in China, that is a recombinant virus that might have entered from the Russia to China, that is having different genetic background than the virus we are having. So this is an ancestral African strain, that is Kenyan strain. So in 2020, it has spread to other parts of most of the countries of the Southeast Asia. And during this time also, it spread to Sri Lanka, Taiwan, and other countries. And recently in 2022, because surprisingly earlier, there was no report from Pakistan. So it is January, it created a havoc in Pakistan, 2022, January. So, but the exact origin of the virus is still not known, because they have not reported the genetic characterization of the virus completely as of now. So in India, we got the first outbreak in 5 coastal districts of Odisha in August 2019, and it was notified to the OIE, World Organization of Animal Health, by November, after the first isolation of the virus and differential diagnosis carried out at the NISA. And the 2000, as everybody is aware, 2022 LSD outbreaks, they have been devastating with large number of cattle deaths in several states. Next one, please. Next slide. So this was the before 2000, around 2005 to 2020. This was the geographic expansion around the world. So most of the disease was restricted and it was endemic to African countries, followed by the Middle East, mostly Israel. Israel was the
Mishra,
Turkey. Yes, sir.
we would like to listen more about diagnosis, sir. Please.
Yes, sir. I will just skip. I'll go fast.
Yeah.
Next one. Next. Next one. Already host range. Everybody knows this is the predominant host is cattle, and sometimes it occurs in buffalo. But recent reports of natural infection and death in captive giraffe in Vietnam, they have shown this it has caused death also in captive giraffe. And what we have seen also, there are some sporadic reports of wildlife LSD in wildlife in Rajasthan also, recently in 2022. But everybody, as we are aware, most of the serological evidence in wild ruminants and epidemiology in wild ruminants is not definitively known, and it is not genetic. Next one. So the transmission already, I think Dr. Gulati has initially described. The principal method is mechanical transmission. And it is now known, recent evidence shows that this Stomachsis calcistrans is the major vector which has been identified in Europe. But in India, we have not shown what is the exact vector which is producing. And they have shown that it is retaining the virus in proboscis of vector for 6 days in the proboscis of the Stomachsis calcitrans. And minor source is direct contact or ingestion of contaminated feed and water. But now the emerging evidence, because this recombinant strain which is circulating in all of the Southeast Asia, they have shown that it is spreading through more by direct contact and indirect contact, then also simultaneously along with the mechanical vector transmission. So we have to be very careful if any entry occurs, this recombinant strain from other countries. So this may be the other routes of transmission also, we have to be very careful. And the persistence in nature, it is not definitively known, but viremic asymptomatic cattle, they are suspected. And it is highly likely that the cattle which are showing generalized nodular lesions, they are the most important source of infection. So we have to control whenever there is any generalized infection happens in cattle. So we have to be very careful. The most of the transmission occurs from there. Next one, please. So I'll just go briefly because it is already known. It is having a 150 kilobase pair genome, having 156 putative genes. But my focus of point is it is phylogenetically distinct with two main lineages. One is wild type strain, second one is the vaccine strain. And among the wild type strains, so there are two lineages, two sub-lineages, because one is the ancestral Kenyan strain, second one is the European strain or Middle East, Europe, and also occurs in Africa now. So we have to be, because there is a deletion in this 12 nucleotide deletion in this, the virus which is circulating in Middle East, Europe, and Africa. But in ancestral strain, this virus, this deletion is not there, nor in the vaccine strain. So in now, whatever the vaccine strain we are having in India, recent LSD vaccine strain, that also has this GPCR. This is not having the deletion. It is having that full GPCR gene. And again, vaccine strains and recombinant strains. Now they have become on the basis of genetics. There are 4 clusters within recombinant strains. Next one, please. Yeah. So coming to this genomic organization, this is the broad picture view. Already I have described what is the lineages, sub-lineages. Next one, please. Yeah, coming to this diagnosis, which is the focus, because this is the why prescribed diagnostic tests. So these are the agent identification, so virus isolation, gold standard test for fast identification in a country, and primary LT cells or lamb kidney cells are recommended for preliminary isolation. Afterwards, the MDBK cells and virus cells can be used for culture and growth. But what I have seen that we have seen most of the cases from field cases also, they grow better in LT cells rather than MDBK or Biro cells. Afterwards, once they are optimized in LT cells, further passaging is possible in those cells. So there are, in this virus isolation, several people use different type of cells. Of course, now it is recommended both MDBK and Biro cells can also be used. And the genome detection is mostly done by the PCR, that is a Capripox generic PCR, because it is known that there is no sheeppox, goatpox infection, virus infection in cattle. So easily it can be used in cattle, this PCR, Capripox generic PCR, which is targeting the P32 gene. And it is a recommended primers are there. And real-time PCR also can Pan-Capripox Stackman PCR. So initially it can identify all the strains, whether it is vaccine or these wild type strains. But afterwards we need the differentiation that is possible through DIVA-Q PCR test. And for serology, as everybody knows, it is a single serotype. When all the Capripox viruses, they cannot be differentiated by serology. So this is mostly it is virus neutralization test and the ELISA. Now the commercial ELISA is available only from one source, that is Capripox double antigen ELISA. So it is used, it can identify both antibodies against sheeppox, goatpox as well as LSDB, but it is commercially available only for a single source now. Next one, please. So briefly, I will just skip this one because already our previous speakers, they have described. But eradication, there are two methods, stamping out or vaccination. So most of the European countries are where the people, it is possible, so they go for stamping out or combination with stamping out or vaccination. But as you know, in India, it is not possible any stamping out. So vaccination is the only choice. And the live attenuated homologous vaccine is the choice as for the WH. But the critical hallmarks already, I think it has been discussed during Dr. Gulati's talk, benefits of vaccination. But there are certain drawbacks, vaccination, long waiting period to achieve LSD-free status, adverse events like needling disease, as seen in other countries. But in our case, what Dr. Naveen Kumar has shown, it is not having that deletion, 126, LSD 126 gene deletion. Usually it is a marker for the LSD needling vaccine strain, which is not shown in whatever the LSD vaccine strain we have been produced in India. And generation of recombinant LSDV following live attenuated vaccine. Next one. This I will skip because already it has been discussed. Vaccines. Next. So I'll just give a brief overview because this initial outbreak was attended by ICAR-NIVEDI and scientists from IVRI. And after the fast isolation, identification, differentiation, we submitted the report to DASD. Followed by it was then.
That Mishra,
Yes, yeah.
that Mishra, we are already late, so I would request kindly you. I think you have already covered your diagnostic part.
No, sir, I am coming. Just I will go today. Now focus. I will just next slide.
So come directly to the diagnostic. Yeah. Don't go in this history because most of the people they already have read the paper.
Yeah. Next one, please. Next one. Next slide. Here, sir, I will just try to make up this genetic phenology analysis. What we have done currently circulating LSD strains in India. They are closely ancestral Kenyan strains. But recently in 2022, what we have seen that some mutations are occurring also in this virus from the 2022 outbreaks from Rajasthan and Himachal Pradesh also. Other sequencing studies are underway because it is showing some deviation from the earlier ancestral cell. So next one. So this is the picture of the earlier till 2021, 19, 20, 21. So this was the virus circulating in the ancestral strain. Next one. And this is the situation in 2020 till December. So now the affected states, the states are this year it was affected 20 states and the past report in 7 states, all the red color they have shown this was past confirmed report. Next one, please. And this is the status in till 2022, 27 states and union territories. We got this Andaman Nicobar this year, Jammu and Kashmir, then Chandigarh also, few of the, which was earlier not this one. And interestingly, from Ladakh, we have not received any samples. So this Ladakh region is still free from LSD as of now. Next one, please. So coming to this, the current strategies of the government of India. Now this national strategy, the advisory and guidelines on LSD diagnosis and control issued by government of India to all the states in November 2019. So all states to carry out LSD clinical surveillance, followed by cattle and buffalo samples from suspected cases to ICAR-NIVEDI for laboratory testing and confirmation as per NIVEDI sampling guidelines. And once LSD is confirmed in a district, further testing at DASD designated LSD testing labs in states. So now in 2022, due to the vast number of outbreaks around the country, so currently there are 32 designated LSD testing labs. Recently DASD has authorized. So now once we confirm a disease in a state, so rest of the things, the state level laboratories, they can go ahead. NISAD and other testing laboratories send result to Government of India. And Government of India notifies LSD occurrence to the concerned state for undertaking prevention and control measures. And the state and government undertakes implementation of the LSD prevention and control measures. Next one, please. So these current methods of LSD diagnosis, what we are doing once this molecular test, we are mostly carrying out the molecular test by viral genome detection by real-time PCR. So first is we are doing the P32, that is a generic Capriparous generic, and simultaneously we are focusing on this because these primers are recommended primers by because in these states, many of the states, they don't have the real-time facility. So we have supplied to them both the diagnostic PCR as well as diagnostic real-time PCR. So many of the states laboratories where this facility is not there and real-time PCR is not available, they can go ahead with this diagnostic PCR. And wherever it is available, this real-time PCR, so they can straight away directly go by our this real-time PCR, which has been optimized, and we have seen that it is working nicely since last 3 years. But some deviation now occurring due to mutations, we have to update this data on this real-time PCR primers and probe also. Next one, please. Coming to this LSD specific real-time PCR, what we are using this targeting this LSDV 126. So that is the external envelope virus gene, EEV gene that is targeted. Because peculiarly this is the only reason we are having in wild type strains 2 copies of this gene. Whereas in vaccine strain like Nithling strain, it is having only 1 copy. So that means the complete deletion of this gene 126, 1 copy in all Nithling. So that's why current real-time PCR DIVA tests are based on this means 126 targeted. Because we can directly tell that it is whether it is a wild type strains or a vaccine strain, Nithling vaccine strain. Of course, now with the recent development of vaccine at NRC, so and Dr. Naveen Kumar presented the another region, ITR region, this deletion because that is not happening in 126 gene. So again, that has to be refined and tested, and again we have to see this how it is the optimized and validity of this qPCR whenever it is implemented for differentiation after the between the vaccinated animals and the infected animals. And what we do is initially if we get some suspicion also, so this differentiation with other strains, other wild type strains, because in Europe whatever the wild type strain is circulating, that can be differentiated by a GPCR, DIVA assay, QRT-PCR, because both will be produced. They have both these genes, 126 duplication in both the wild type sublineages, but whereas in GPCR there is a deletion. So there is this target genes for genetic characterization. We usually go for GPCR, RPO30, EEV, or whole genome sequence. So already I have told this qPCR wild type strains and Nithling vaccine strains because that will be very much required once we start vaccination. And the critical observation what we have seen is preferred sample types is skin nodular tissue is the best sample, followed by nasal swab or blood or semen. And one thing I want to just point out here. So even after recently we have seen in Punjab, some of the bull semen farms, semen bull station farms, they are shedding the virus even after 3 months or 4 months. Still they are shedding some of the bulls. So we have to be very careful because once he infected this frozen semen bull, these production facilities, so they means we have to test for the shedding also. So we have to be very careful. Next one. And one more important point is differential diagnosis. What we have recently observed that in mild cases of buffalo in several countries, because already, as you know, earlier we have been this already buffalo pox, cow pox, especially buffalo pox outbreaks has been occurring in most of the several states. So we have to differentiate this differential diagnosis by pseudo LSD, bovine herpes virus, buffalo pox, cowpox, pseudocowpox, and buffalo papillary stomatitis. Especially in buffaloes, this differentiation between buffalo pox as well as LSD. Because mild cases of LSD, many samples we are getting, they are negative. Maybe they may be due to different, either it is a buffalo pox or some other disease. And the diagnostic challenges still remain. The sampling is the most problematic because the correct sampling at the correct time and the vials, many people, they do not even have a proper vial in state facilities. And this is straight infrastructure, that is very important. Otherwise, it will produce all the laboratory tests, it cannot be confirmed. And sample labeling, clinical history, they are very much important for sending the samples, testing the samples, and harmonization of the test. That is very much crucial. Among all the laboratories, the harmonization of the test should take place. Otherwise, there will be discrepancy in the results. Next one, please. So these are some of the photographs which already we carry out here in differential diagnosis with the different, all the whatever the differential diagnosis we have been carried out. Next one. So coming to the conclusions and future perspective, LSD emerged in 2019 and it has spread. It is spreading and evolving in throughout the India. But the source of origin not clearly known, because we know that it is ancestral virus, but the same virus is also circulating in Bangladesh as well as in Nepal and Myanmar. Recently, it has been shown the virus circulating in India, Nepal, Bangladesh, and Myanmar, they belong to the same virus. Whereas in China and other Southeast Asian countries, they are different viruses. They are all recombinant viruses. So we have to be very careful. Anytime these recombinant viruses come, so there will be a challenge in diagnosis, differentiation. Basically, it cannot be differentiated. They are having this wild type strain backbone, along with that vaccine strain. So there is a recombination at different places. So that is not a unique deletion or mutation you will find. So it will be a very much challenge in diagnosis. This wild type, and again there is a risk of introduction from wild type strain from Middle East, because there is a already we are having ancestral Kenyan strains. So that may also likely to occur. And the high impact on cattle production and livelihood of dairy farmers, epidemiology and transmission dynamics need to be studied in detail. That is still lacking. And challenges in diagnosis and strain differentiation. There is a need for refinement in national policy on vaccination strategies, awareness of field vets and farmers on LSD, and the implementation, correct implementation of the prevention control measures, especially now is the time to vaccinate. Because if we vaccinate by February, March, so then the disease incidence will be less during the monsoon season. Otherwise, in each year, we get thousands of samples flooded to different laboratories for confirmatory diagnosis. Thank you all.
Thank you, Mr. I'm sorry I had to interrupt you 3 times, but because I'm paucity of time, so I'm sorry for interrupting you. So, colleagues, now we move on to next speaker, Professor Y.S. Malik. Before I invite Professor Malik, I would like to request Dr. Madri for a brief introduction. Dr. Madri.
Thank you, sir. Welcome, Dr. Malik. He joined a bit late, probably when we started. He was not available to finish the activities in college. And I will introduce Dr. Malik. He has been currently serving as Dean Veterinary College of Animal Biotechnology. Sorry? Previously, he has served as ICAR National Fellow at ICAR-NIVEDI, and then he is having a very rich experience in the field of virology and biotechnology, and his major research achievements include contribution to viral disease epidemiology, virus host interactions, microbial biodiversity, characterization and diagnosis of animal pathogens. And basically, he has worked on coronaviruses, Japanese encephalitis virus, rotaviruses, and many other zoonotic viruses. He has been a recipient of many prestigious awards, including ICAR Jawaharlal Nehru Award and Young Scientist Awards of Association of Microbiologists in India. And he has been honored with several fellowships, including NAS and NAVS and National Academy of Biological Sciences, National Academy of Dairy Sciences, and many others. And he is a member of ICT, which is a prestigious thing, on Inaugural and Picona study group. And he is a managing committee member of World Society on Virology, to which he is also a Secretary General, and he is also a Secretary General in IVS. And to tell about him, it is very, very difficult. I think it's quite a long list, and I'll be cutting it short. And he is an associate editor of 20 years in microbiology and 20 years in veterinary sciences. And he has been associated with almost all societies on microbiology and virology in India. And to say that his research contribution, we can say that he has got an index hovering around 50, and citations have over 10,000. I request Dr. Singh to welcome.
Thank you, Dr. Himadri. So now I invite Professor Vaisnath Malik. What do you, Dr. Malik?
Thank you very much, Dr. Himadri, for your good words, and thanks to the organizers of this event, National Workshop Brainstorming that we are having on LSD. I am thankful to the chairs at NIVEDI today, Dr. R.K. Singh, Dr. Bhatta, Dr. Sanyal, and all the learned members who are present there. Briefly, I take this opportunity on behalf of the Indian Virological Society to say thanks to the NIVEDI for taking up this novel cause to spread the message, awareness amongst the society, the stakeholders, and Indian Virology Society is playing this role since 1984 when it was established. Dr. Himadri, I am not able to see my slides.
Yes. Oh, 1 second. Yeah, we are sharing it again. We are sharing it again. It is visible, sir?
It's not visible to me. Or you can allow me to share, then I can share it from this place.
Okay. Yeah, that will do. Yeah. Now,
Yes, can I share now?
is it visible now?
Not visible to me anything.
चलो आप उसमें ही शेयर करें. आप कर दीजिए शेयर.
Please allow me then.
Dr. Malik, you can share now.
Can you see my slides?
Not yet. Yes, yes.
Thank you very much. So I will go straight away with the Indian Virological Society. This is one of the society that exactly we can call as the One Health Society, because we have the plant, we have the medical, we have the fisheries virologist in this, along with the animal virologists. So this this society came in existence in December 1984 at HAU ISAR, and now its headquarters is at Delhi, and we are in the NAS complex. Our official headquarters is there. So we have the uniqueness that I told, that we are the experts from different areas are there in this society. And basically this society, what we are organizing today as the seminar workshop, we are organizing continuously under the aegis of this society. Several international conferences have been organized, workshops, and academia industry meet. And we are partner of the International Union for Microbiological Society, World Society for Virology, that is one of the growing society world over, with over 1990 countries are the partner of this. This society also publishes a journal that through the Springer Nature, that is the virus disease. And good thing to note from this virus disease is that the papers are over 32 countries, they have been published in past in this journal. And it is in Scopus and several indexing agencies. The society is having more than 500 active members. And right now we are honored to have our president, Dr. R.K. Singh, and who is the chair of today's function also. And we have different sections like plant virology, medical virology, then all the secretaries and vice president in different sections we are having, along with the treasurer and one editor-in-chief who is from the medical side in this case. We were honored to have very prestigious personalities guiding us as the president, including Dr. K.S. Bhargava, S.P. Rai Chaudhary, Professor M.P. Yadav, Professor Narayan Rishi, Professor Pradeep Seth, Dr. A.K. Prasad, and our president, immediate past president, Professor Anupam Verma. These are some of the listing, but I was telling that we are continuously organizing the conferences, national as well as the internationals, and recent our conference was at Skost, Kashmir. We have been organizing the workshop and trainings, and this is by Dr. Gulati himself. Last year also he organized under the Zoonosis Day, on that day of Zoonosis, and again he took that responsibility from IBS as a secretary of veterinary side and organizing this seminar on the LSD. So this is the continuous activity, and next month we are organizing one more at the Guwahati at Khanapara, that is on the transboundary diseases. These are some of the glimpses from various activities what we are organizing. This is from the Skost, Kashmir. Then our previous activities at Hyderabad, AIMS Hyderabad, that we organized last year. These are from the INSA. We organized one international conference attended by more than 30 international speakers. This is at the PGI Chandigarh, and several other that we have, even we have gone to the northeastern state, Shillong. This is the journal I was talking about, that it was earlier Indian Journal of Virology, but now it is named as the virus disease since 2014, and it is having the site score of 3.5, and we are hoping to see that it will get a very good Thomson-Reuters impact factor very soon. Our different experts are publishing the books under the banner of Indian Virological Society. And these are some of the best books which have been considered world over on the corona, plant virology, emerging transboundary, that Dr. R.K. Singh and myself and Dr. Yadav, we published this from Springer, recent advances in animal virology also. These are some of the awards what the society is recognizing the different experts in the field of virology. We support the youngsters by travel grant, even the Young Scientist Award, provide the fellowships to the extraordinary performing people. Then the senior most awards are Professor K.S. Bhargava's and the Syama Prasad Rai Chaudhary Award. So this is all about the Indian Virological Society. And again, I thank the organizers of this event for taking up this event under the aegis of Indian Virological Society. Thank you very much, Dr. Gulati. Briefly, I will go with the Punjab status of LSD, because today's discussion yes,
full screen में कर लीजिए presentation को. Yes, sir. Good, thank you. Thank you.
So this is basically the data that has accumulated at the Punjab State Animal Husbandry that I am presenting, being a part of that meetings what I am attending with the government of Punjab. So our Punjab is at the northern state and well known for its cultural and agricultural activities. So Punjab is divided into three parts. One is the upper side left, that is the Doaba, Majha, and the Malwa region. Malwa is the largest region where the Gadwasu, our university, is located in Ludhiana. And this is the total livestock species that is distributed in Punjab. So basically, if we go with this picture that shows that majority of the animal species is cattle and buffalo, and very small population is of the sheep and goat, and pig is very small, nearly 45,000 only. So majority of the population is cattle and buffalo, and both were the most susceptible species considered species for the LSD that affected the Punjab also. The cattle population, if I go, some of the regions are well populated by the cattle, and this is just one description of that population scattered through different 3 regions of the Punjab. Majority of the cattle population is Holstein HF crossbred, and only few are the specific Jersey or Jersey Haryana and other breeds, Sahiwal. In the buffalo, the population is highest, nearly 45 million population of the buffalo is in Punjab, and majority of the population in buffaloes is of the Murrah and Murrah graded buffaloes. This is about the LSD in Punjab. We were affected. First time the case came in July week, first week of the July, and later on then cases started appearing in different parts of the Punjab. And as of 10th November, after 10th November, we are not getting any of the case for the LSD that is coming positive. And as of now, the animals total they have been affected is 1.75 lakh. And out of that 1.27 lakhs, the cattle have recovered. And what is the good thing is that the vaccination was done at a very, very large scale. And within a month, nearly 9 lakh vaccinations were done throughout the state. And the bad thing that happened is the loss of our cattle. Nearly 17,932 cattle, they were reported to, during this period of the outbreak, they died because of the LSD, or they were suspected for the LSD. This is the status of the vaccination in different districts of the Punjab. And highest vaccination was done in the Ludhiana region. Nearly 1 lakh cattle, they were vaccinated with the LSD vaccine. And majority of the vaccine, or almost all vaccination, what has been done is of the goat pox vaccination. This is the what I was telling, that Ludhiana is at the highest. It is 89,000 vaccinations they were done within 1 to 2 months. And this is the status of the affected, recovered, and deaths which were reported. So highest affected cases were also in Ludhiana and Moga, and that's why the maximum coverage was given for the vaccination also in this area. And mortality was highest in Bathinda, Ludhiana, and Moga. This is again the same thing has been detailed in this case. Cattle population that was affected badly by LSD is in the Patiala, nearly 31,000. Then because another species that was at the risk was the buffalo, but cases which were found in the Punjab are very limited. Only 138 cases came for the having suspicion for the LSD. And out of that 63 they recovered, and 8 animals they were reported died in Ludhiana because of the LSD. This is the vaccination status. In Gausala there was a hue and cry, and huge mortality was also seen in case of the Gausalas, and that's why one focus was given that within a month 57,000 animals were affected in the Gausalas. And the coverage was nearly 1.17 lakh. But some of the Gausalas, they use their own resources other than the government also. They purchase the vaccine, and they have gone with the vaccination of the Gausala animals. So this is about the same affected, vaccinated by some of the own Gausalas. and maximum of affected animals are again from the Patiala in the Gausala. That region is having maximum Gausalas. This is the one thing that government of India, because so many cases were coming and samples were sent to the Nishad Bhopal. So in addition to that, government of India, they approved Gadwasu College of Biotechnology as the testing facility for the LSD. The first case will be confirmed by the Bhopal, and following the cases in the same district, they will be screened by the Gadwasu. As of now, we have received some samples from blood, even semen, what Dr. Niranjan was also telling, and different states what have been assigned to Gadwasu or the Ludhiana is the Punjab, J&K, Ladakh and Chandigarh. So majority of the samples what we have received is only from the Punjab region. And this is one appeal that has been submitted by the government of Punjab through our Principal Secretary Animal Husbandry, Vikas ji, to government of India that this LSD can be included to be included as the NADCP under the NADCP for vaccine availability. So this is all about from Punjab and about the IVS. I'm thankful to each and everyone who is present and who are virtually they are associated with this event. Thank you very much.
Thank you, Dr. Malik, for an excellent presentation, finishing in time, both and covering both IVF and also your presentation. We are very happy. Thank you so much.
Thank you so much, sir.
Now we have our final presenter, Dr. Amit Kumar. Before I invite Dr. Amit, I request once again Dr. Hemati for a brief introduction of Dr. Amit Kumar.
Thank you, sir. Dr. Amit is the youngest of the speakers we have, and he is working at the Scientific Division of Virology, ICAR, IVRI, and he did his master's and PhD from IVRI, and he has been working on the development of